Welcome back to Sleepy Tin Foil Hat. Settle into wherever you are tonight. Let your shoulders drop. Let your breath find its slower rhythm. We are going to spend the next several hours walking together through one of the most fundamental yet least understood processes that every living creature on this planet experiences. The process of aging. The slow accumulation of changes in our bodies and minds that begins essentially at birth and continues throughout our entire existence, eventually leading to the various deteriorations and ultimate ending that characterize mortal life. Every human who has ever lived has been engaged in this process, and the question of what aging actually is, why it happens, and whether anything can be done about it, has occupied human thought for as long as humans have been thinking. Tonight, we are going to examine aging carefully, with the kind of patient attention that allows the genuine strangeness of this process to become visible. Because aging, when you actually examine it properly, turns out to be far more interesting and far more genuinely puzzling than the casual encounter with it suggests. Before going further, a small favor. If you find this kind of slow, careful exploration helpful for falling asleep, or if some part of your mind genuinely enjoys thinking about the deep features of biological existence, while the rest of you drifts toward unconsciousness, please consider subscribing to Sleepy Tin Foil Hat. The channel grows when listeners signal that they are here, and the algorithm is the only way new people find their way to this kind of patient nighttime thinking. And if you would, leave a quiet note in the comments telling me where in the world you are tonight. The country, the city, the hour on your clock if you want to share it. I read these. There is something genuinely moving about watching the slowly forming map of where this audience is scattered. All of us awake or trying not to be at the same hour, listening to the same voice across many different time zones and many different lives. So tell me where you are and then settle deeper, because the journey from here will be long and patient, and I think worth taking carefully. Now to begin where most discussions of aging begin, which is with the basic question of what aging actually is. The intuitive answer is something like this. Aging is the process of getting older, the accumulation of years lived, the gradual approach toward the eventual ending of life. This intuitive definition captures something, but obscures the genuine complexity of the phenomenon. Because aging is not really just the passage of time. It is a specific biological process with specific mechanisms operating at the molecular and cellular levels, producing specific changes in our bodies that we recognize as the symptoms of growing older. The passage of time itself does not cause aging. The biological processes that occur over time cause aging. The distinction matters because it opens the possibility that the biological processes could, in principle, be modified, slowed, or even reversed without changing the passage of time itself. The history of human thought about aging is itself fascinating. For most of human history, aging was understood as a natural and inevitable feature of existence, similar to other inevitable features like death itself. The various religious and philosophical traditions developed elaborate frameworks for understanding aging within their broader conceptions of human existence, with the process being interpreted variously as a punishment, a test, a preparation for what comes after death, or simply as the natural pattern of mortal life. Various traditions developed practices intended to slow or delay aging, including dietary practices, contemplative practices, and various herbal and medicinal interventions. The Chinese tradition, in particular, developed elaborate practices aimed at longevity, with Taoist alchemists pursuing both internal practices and external substances that were believed to extend life. Similar pursuits occurred in many other traditions, with the universal human desire to remain young and avoid the deteriorations of age driving widespread efforts across cultures and centuries. The modern scientific study of aging began relatively recently, with significant work emerging primarily in the 20th century. Before then, aging was generally accepted as something that simply happened, with limited systematic investigation of its underlying mechanisms. The development of modern biology, particularly molecular biology and cell biology, eventually allowed researchers to begin examining what actually occurs in cells and tissues as organisms age. The findings have been remarkable, and they have transformed how we think about the process of aging in ways that have substantial implications for what might be possible in terms of extending healthy human lifespan. Modern biology has identified what are now called the hallmarks of aging, the specific biological changes that occur in cells and tissues as organisms grow older. These include changes at the genetic level, with mutations accumulating in DNA over time, and various changes occurring in the patterns of gene expression. They include changes at the level of the structures that protect chromosomes, with the telomeres at the ends of chromosomes shortening with each cell division, and eventually limiting how many times cells can divide. They include changes in the proteins that perform essential functions in cells, with damaged proteins accumulating over time, and the systems that normally clear away damaged proteins becoming less effective. They include changes in the structures within cells that produce energy, with mitochondrial function declining and contributing to various deteriorations. They include changes in how cells respond to stress and damage, with the various repair mechanisms that maintain cellular function becoming less efficient with age. They include changes in the populations of stem cells that produce new cells to replace those that die, with stem cell function declining and contributing to the inability of aged tissues to repair themselves as effectively as young tissues do. These various changes do not occur independently of each other. They interact in complex ways, with damage in one system contributing to dysfunction in others, and the cumulative effects producing the patterns of decline that we recognize as aging. The complexity of the interactions has made the study of aging genuinely difficult, with researchers having to map out not just individual mechanisms, but also the relationships among them. The picture that has emerged in recent decades is of aging as a complex multifactorial process, with multiple interconnected mechanisms operating together to produce the overall trajectory of decline that all living organisms experience. The strangeness of this picture, when you actually sit with it, is worth examining carefully. Aging is not a single thing. It is a constellation of related processes that together produce the patterns we recognize. Each of the individual processes can, in principle, be studied separately and potentially modified separately. The accumulating evidence from research on various organisms suggests that interventions targeting individual mechanisms can sometimes have substantial effects on the overall pace of aging, even though the interventions only directly affect one component of the larger process. This has opened up the possibility of interventions in human aging that could potentially extend healthy lifespan by modifying specific underlying mechanisms. Tonight, across the parts that follow, the exploration will work through more of the strange and genuinely interesting aspects of aging. The various theories about why aging happens at all, with the evolutionary biology providing some surprising answers. The specific mechanisms that have been identified at the cellular and molecular levels. The remarkable diversity of aging patterns across different species, with some organisms showing essentially no signs of aging, while others age extremely rapidly. The various interventions that have been shown to extend lifespan in laboratory organisms, and the questions about whether similar interventions might work in humans. The differences between chronological age and biological age, with the recognition that two people of the same calendar age can have very different biological ages. The questions about whether human lifespan has any specific upper limit, or whether the current observed maximum reflects only the limits of current technology rather than some fundamental biological constraint. The various social and psychological dimensions of aging, with the experience of growing older being shaped by cultural and individual factors in addition to biological ones. and the genuinely deep questions about what aging means and whether efforts to extend lifespan are wise or unwise. What is not part of this exploration is simple advocacy for any particular intervention or any particular position on the desirability of life extension. The science is what it is. The various interventions that have been investigated have produced varying results. The implications of potentially extending human lifespan involve substantial complexities that deserve honest acknowledgement. The careful examination of aging is more interesting than simple advocacy, because the actual situation is more complicated and more genuinely strange than either pro-longevity or accepting of mortality simplifications can capture. What to hold in mind as the journey settles into its rhythm is that you are engaged, right now, in the same process of aging that every living human is engaged in. Your cells are accumulating various forms of damage as you listen. Your telomeres are slightly shorter than they were at the beginning of this audio. Your proteins are slightly more damaged. Your various biological systems are slightly older. The process is continuous, with each moment of existence involving some increment of the larger trajectory. You did not choose to be involved in this process, and you have limited ability to alter it through individual decisions. But you are nonetheless its subject in every moment of your existence, with the cumulative effects building up over the years and decades of your life. The contemplation of this process, on a quiet night like this one, is itself a kind of acknowledgement of the deep biological reality that connects you to every other living being that has ever existed. Lower the lights, if you have not already. Let your awareness soften. The process to be examined is one that has been operating in living organisms for hundreds of millions of years, with countless different species having evolved their own specific patterns of aging through the long history of life on Earth. The relationship between us and this process is one of the most fundamental aspects of what it means to be a living creature. Tonight, the careful contemplation of this relationship will be one of the small ways to engage thoughtfully with our own biological existence in the patient way that suits a long, quiet evening. The question of why aging exists at all is more puzzling than it might initially seem. From a casual perspective, aging seems obviously natural. Everything wears out eventually. Cars break down with use. Buildings crumble with time. Living things, similarly, must inevitably deteriorate as they accumulate wear from the activities of living. This intuitive view treats aging as a kind of mechanical wearing out, with the deterioration being an inevitable consequence of the passage of time and the activities of existence. But this intuitive view fails on closer examination. Aging is not simply mechanical wear and tear. It is a biological process, and biological systems are remarkable for their ability to repair and renew themselves. Your body, right now, is continuously producing new cells to replace old ones, repairing damaged tissues, removing damaged molecules, and maintaining the various systems that allow you to function. The repair and maintenance processes that operate within living organisms are extraordinarily sophisticated, far exceeding what any human-built machine can accomplish. If biological systems are this good at maintenance and repair, why do they nonetheless age and eventually fail? The question is genuinely interesting, and the answer turns out to involve some surprising features of evolutionary biology. The evolutionary explanation for aging begins with a simple observation. Natural selection acts most strongly on traits that affect reproduction. Organisms that reproduce more successfully pass on more of their genes to subsequent generations, and the genetic variants that contribute to reproductive success become more common in the population over time. Organisms that fail to reproduce, whatever the reason, do not pass on their genes, and the genetic variants associated with reproductive failure become less common. The cumulative effect over generations is that populations become genetically optimized for reproductive success. The crucial point is that natural selection acts most strongly during the reproductive years of an organism's life. Genes that affect health and survival during youth and middle age, when reproduction is occurring, are under strong selection. Genes that affect health and survival only at older ages, after reproduction has typically ceased, are under much weaker selection. The reduced selective pressure at older ages means that genetic variants that would cause problems late in life, but that have no effects during the reproductive years, can persist in populations because natural selection cannot effectively remove them. The accumulation of these late-acting genetic effects across many generations is one of the major proposed explanations for why aging exists. This explanation, developed initially by various evolutionary biologists in the 20th century, has several specific versions. The mutation accumulation theory proposes that aging results from the accumulation of genetic variants that have negative effects only late in life. Such variants escape selection because they do not reduce reproductive success, and they accumulate in populations over evolutionary time. The antagonistic pleiotropy theory proposes that some genetic variants have beneficial effects during youth and harmful effects during old age, with selection favoring the variants because the benefits outweigh the costs from a reproductive standpoint, even though the harmful late-life effects contribute to aging. The disposable soma theory proposes that organisms face trade-offs between investing in reproduction and investing in long-term body maintenance, with the optimal balance from a reproductive standpoint involving relatively limited investment in maintenance, leading to gradual deterioration of the body over time. These theories are not mutually exclusive, and the actual evolutionary explanation for aging probably involves elements of all of them operating together. The cumulative picture is that aging exists because natural selection has not strongly favored organisms that maintain themselves indefinitely. The selective pressures that have shaped living organisms have prioritized reproduction over maintenance, with the result that bodies are optimized for successful reproduction rather than for indefinite survival. The aging we observe is therefore not a failure of biology, but a reflection of the priorities that have shaped biological systems through evolution. This perspective has implications worth sitting with carefully. The aging that we experience is not necessary in any absolute sense. It is the result of specific evolutionary processes that have produced the kinds of organisms we are, with the trade-offs and limitations that those processes imply. If the selective pressures had been different, the kinds of organisms that exist would also be different, potentially including organisms that age very differently from how we do. The biology that produces our aging is, in some sense, contingent on the particular evolutionary history that has shaped our species, rather than being a necessary feature of life itself. The diversity of aging patterns across different species supports this perspective dramatically. Different species age in remarkably different ways, with the variations reflecting the different evolutionary pressures that have shaped them. Some species age very rapidly. Various small organisms, including many insects, have lifespans of just weeks or months, with rapid maturation followed by rapid aging and death. Other species age very slowly. Various tortoises can live for over a century, with relatively slow aging throughout their lives. Some species appear to show essentially no signs of aging at all. The hydra, a small freshwater organism, has been studied extensively and shows what biologists call negligible senescence, meaning that older hydras do not show the patterns of declining function that characterize aging in most other species. The hydra's ability to continuously regenerate from its stem cells appears to allow it to maintain its biological function indefinitely, with the only causes of death being external factors like predation or environmental stress. The bristlecone pine trees that grow in high mountains of the western United States provide another remarkable example. The oldest known bristlecone pines are over 4,800 years old, with these ancient individuals continuing to grow and reproduce despite their extreme age. They show some signs of aging, with damage accumulating in various ways, but they continue to function essentially indefinitely under appropriate environmental conditions. The biology of these ancient trees has been studied extensively, with researchers trying to understand how they managed to persist for such extraordinary spans of time. Various marine organisms also show remarkable longevity. The ocean quahog, a type of clam, can live for over 500 years, with some individuals having been confirmed at over 507 years of age. The Greenland shark is now estimated to live for several hundred years, possibly up to 400 years or more, making it the longest-lived vertebrate species known. The bowhead whale appears to live for over 200 years in some cases. These long-lived species have developed various biological strategies that allow them to avoid or minimize the deteriorations that affect more rapidly aging species. The diversity of these patterns shows that aging is not a single inevitable phenomenon. It is a biological process that has evolved differently in different lineages, with the specific patterns reflecting the specific evolutionary histories of different species. The fact that some species can essentially avoid aging or can dramatically slow it demonstrates that the biology is more flexible than the human experience of aging might suggest. The patterns that characterize human aging are products of our specific evolutionary history, and they could in principle be different if our biology had developed under different selective pressures. The implications for thinking about potential interventions in human aging are interesting. If aging is not a single inevitable process, but a complex biological phenomenon that has evolved differently in different lineages, then the possibility of modifying human aging through deliberate intervention is not as absurd as it might initially seem. The various biological mechanisms that contribute to human aging are specific processes that can be studied and potentially altered, rather than mysterious forces that operate beyond the reach of investigation. The biological diversity of aging patterns across species suggests that there is no fundamental barrier to substantial modifications of aging, even though the specific interventions required to achieve such modifications in humans remain to be developed. The molecular mechanisms that have been identified as contributors to aging provide specific targets for potential intervention. The shortening of telomeres can potentially be addressed through interventions that maintain or restore telomere length. The accumulation of damaged proteins can potentially be addressed through interventions that enhance the cellular processes that clear away damaged proteins. The decline in mitochondrial function can potentially be addressed through interventions that support mitochondrial health or replace damaged mitochondria. The depletion of stem cells can potentially be addressed through interventions that support stem cell populations or that introduce new stem cells. Each of these mechanisms is being actively investigated by researchers who are developing potential interventions, with various approaches showing promise in laboratory studies, even if their applicability to human aging is not yet fully established. The research on caloric restriction is among the most well-established findings in the biology of aging. Studies in various organisms have consistently shown that reducing food intake below the levels that the organisms would normally consume can substantially extend their lifespans, often by 20 to 50% or more. The effect has been demonstrated in yeast, worms, flies, fish, mice, rats, and various other species. The mechanisms by which caloric restriction extends lifespan involve multiple pathways related to cellular stress responses, repair processes, and various aspects of metabolic regulation. Whether the effect occurs in humans is not entirely clear, since the long human lifespan makes definitive studies difficult, but various indirect evidence suggests that some lifespan extension may occur in humans who maintain restricted diets compared to those who eat conventionally. The drugs that mimic some of the effects of caloric restriction have been studied extensively, with some showing promising effects in laboratory organisms. Rapamycin, a drug originally developed for organ transplant patients, has been shown to extend lifespan in mice when administered later in life, with the effect being substantial enough to attract considerable scientific interest. Metformin, a drug commonly used to treat diabetes, has shown some lifespan extending effects in laboratory organisms and is being investigated for similar effects in humans. Various other compounds are being studied for their potential to influence aging pathways, with some showing promising results in preliminary studies. The senolytics, drugs that selectively kill aging cells called senescent cells, represent another line of investigation. Senescent cells are cells that have stopped dividing but have not died, instead persisting in tissues where they secrete various inflammatory substances that contribute to various age-related conditions. The selective removal of senescent cells has been shown to extend lifespan and improve various age-related conditions in mouse studies. Whether this approach will work in humans is being actively investigated, with clinical trials currently in progress for various senolytic compounds. The genetic studies of long-lived humans have identified various genetic variants that appear to contribute to exceptional longevity. Studies of centenarians, people who live to over 100 years, have found various genetic factors that may protect against age-related diseases, or that may slow the underlying aging processes. The specific contributions of different genetic variants are still being worked out, but the overall picture suggests that genetics plays a substantial role in determining longevity, with variants that protect against various diseases being found more commonly in people who reach extreme old age. Stretching out before you now, picture the vast diversity of aging patterns across living species. The mayfly that lives only a single day, the bristlecone pine that has been growing for over 4,000 years, the hydra that may not age at all, the Greenland shark that has been swimming through Arctic waters for centuries, the various other organisms that age at their specific rates determined by their specific evolutionary histories. Each species has its own pattern, its own trajectory, its own relationship with time. The patterns we recognize as aging are not universal features of life, but specific outcomes of specific biological histories. The biology is genuinely diverse, with the various possibilities being far broader than the human experience alone would suggest. Tonight, like every night, all these various species continue to live in their various ways, aging at their various rates, experiencing time in the patterns that their biology produces. The diversity is one of the more remarkable features of life on this planet, and the contemplation of it is itself a kind of expansion of how we think about our own situation as one specific instance of the many possible ways that biological organisms can exist in time. The cellular and molecular machinery that produces aging deserves careful examination, because understanding these specific mechanisms is essential to understanding what aging actually is and what might be possible in terms of modifying it. The work that has identified these mechanisms represents some of the most genuinely impressive achievements of modern biology, with researchers having mapped out in considerable detail the specific processes that occur within cells as organisms grow older. Cellular senescence is one of the most important mechanisms that has been identified. Cellular senescence is a state that cells enter when they experience various forms of damage or stress that prevent them from continuing to divide normally. The senescent cells do not die, but they also do not function normally. They accumulate in tissues over time, with their numbers increasing as organisms age. The senescent cells secrete various molecules that affect the surrounding tissue, including inflammatory signals, growth factors, and various other compounds. The cumulative effect of these secretions, which scientists call the senescence-associated secretory phenotype, contributes to many of the patterns of decline that we recognize as aging. The role of senescent cells in aging has become one of the most active areas of research in recent decades. The senescent cells were originally identified as a kind of dysfunctional state that some cells entered, but the broader implications of their presence took longer to recognize. Studies in mice demonstrated that selectively eliminating senescent cells could substantially improve various age-related conditions and even extend lifespan, even when the elimination occurred in already aged animals. The implications for potential human interventions are significant, with various drugs called senolytics being developed that specifically target senescent cells for elimination while leaving healthy cells unaffected. The mechanism by which cellular senescence develops involves several specific pathways. One of the most important involves the telomeres, the protective structures at the ends of chromosomes. Each time a cell divides, the telomeres shorten slightly, with the shortening being a consequence of the way that DNA replication occurs. When telomeres become too short, the cell can no longer divide and enters senescence. The discovery of this mechanism, primarily through the work of Elizabeth Blackburn, Carol Greider, and Jack Szostak, who received the Nobel Prize in 2009 for their contributions, represented one of the major developments in the modern understanding of aging. The telomere story is genuinely fascinating in its details. Telomeres are structures composed of repeating DNA sequences that protect the ends of chromosomes from various forms of damage. Without the protection that telomeres provide, the ends of chromosomes would be recognized by cellular repair mechanisms as broken DNA, leading to inappropriate repair attempts that could damage the genome. The protection that telomeres provide is essential for normal cellular function, but the protection is gradually depleted as cells divide. The progressive shortening of telomeres essentially places a limit on how many times a cell can divide, with the limit being known as the Hayflick limit, after the researcher Leonard Hayflick, who first identified it. The cells of different species have different numbers of telomeric repeats, with some species having much longer telomeres than others. The relationship between telomere length and lifespan is complex, with various studies showing different patterns in different species. In humans, telomere length appears to be related to aging in various ways, with shorter telomeres being associated with various age-related conditions and with mortality risk. Whether deliberate maintenance of telomere length would extend healthy human lifespan is a question that has been investigated but not definitively answered. Some studies have shown that activating the enzyme telomerase, which can extend telomeres, has beneficial effects in laboratory organisms. However, there are concerns about whether such interventions might also increase cancer risk, since cancer cells frequently activate telomerase to enable their uncontrolled division. The accumulation of damaged proteins represents another major mechanism that contributes to aging. Cells contain various systems for maintaining the quality of their proteins, including molecular chaperones that help proteins fold correctly, various proteases that break down damaged proteins, and the autophagy system that clears away larger protein aggregates and damaged cellular components. These protein quality control systems work continuously to maintain the proper function of cells, but they become less effective with age. The result is that damaged proteins accumulate in aged cells, with various dysfunctional protein aggregates contributing to age-related conditions. The most dramatic examples of protein aggregation contributing to disease are seen in various neurodegenerative conditions. Alzheimer's disease involves the accumulation of specific protein aggregates, including amyloid plaques and tau tangles in brain tissue. Parkinson's disease involves the accumulation of aggregates of a protein called alpha-synuclein. Huntington's disease involves aggregates of a protein called huntingtin. Each of these conditions shares the basic pattern of protein aggregates accumulating in specific brain regions and contributing to neuronal dysfunction and death. The connection between protein quality control failures and age-related neurodegeneration is one of the major areas of current research, with various interventions being explored that might enhance protein quality control or reduce the accumulation of pathological aggregates. Autophagy, the cellular process that clears away damaged cellular components, deserves specific attention because of its central role in maintaining cellular health. Autophagy operates continuously in cells, with various components being identified for degradation and processed through the autophagy machinery. The process is essential for normal cellular function, with disruptions in autophagy contributing to various diseases including cancer, neurodegeneration, and metabolic disorders. The rate of autophagy declines with age, with the reduced clearance of damaged components contributing to the accumulation of cellular damage that characterizes aged tissues. Various interventions that enhance autophagy have shown beneficial effects in laboratory studies, with potential implications for slowing age-related decline. The mitochondria, the structures within cells that produce energy through the metabolism of nutrients, play a central role in aging through several distinct mechanisms. Mitochondria contain their own small genomes, separate from the main nuclear DNA of cells, and these mitochondrial genomes accumulate mutations over time. The accumulated mutations can reduce mitochondrial function, with the resulting energy deficits contributing to various age-related conditions. Mitochondria also produce reactive oxygen species as byproducts of their normal function, and these reactive molecules can damage various cellular components including DNA, proteins, and lipids. The damage from reactive oxygen species accumulates over time and contributes to the patterns of aging that affect various tissues. The relationship between mitochondrial function and aging has been investigated extensively, with various interventions targeting mitochondrial health being explored. The development of drugs that specifically protect or restore mitochondrial function is an active area of research, with some compounds showing promise in laboratory studies. The mitochondrial transplantation approaches, in which healthy mitochondria are introduced into cells with damaged mitochondria, represent another line of investigation that may eventually have therapeutic applications. Epigenetics provides another important framework for understanding aging at the molecular level. Epigenetic modifications are changes in the way genes are regulated that do not involve changes in the underlying DNA sequence. These modifications include various chemical tags that can be added to DNA itself or to the proteins that package DNA, with the modifications affecting whether genes are expressed and at what levels. The epigenetic patterns of cells change in characteristic ways with age, with the changes affecting the patterns of gene expression and contributing to the functional decline of aged tissues. The development of epigenetic clocks has been one of the more remarkable recent advances in the science of aging. Researchers have identified specific patterns of DNA modification that change predictably with age, with the patterns being so consistent across individuals that the methylation patterns in a sample of cells can be used to estimate the chronological age of the donor with remarkable accuracy. More importantly, the epigenetic clocks can be used to estimate biological age, which may differ from chronological age. People whose biological age, as estimated by epigenetic clocks, is older than their chronological age, tend to have higher mortality risk and higher rates of various age-related diseases. People whose biological age is younger than their chronological age tend to have better health outcomes. The epigenetic clocks have therefore become important tools for studying aging and for evaluating potential interventions that might slow the aging process. The development of these clocks has revealed that biological age can sometimes be reduced by various interventions. Some studies have shown that lifestyle factors, including diet, exercise, sleep, and stress management, can affect epigenetic age in measurable ways. Various potential anti-aging interventions are now being evaluated using epigenetic clocks as outcome measures, providing a way to assess whether interventions actually slow biological aging rather than just affecting various surface indicators. The ability to measure biological age objectively, separate from chronological age, has been transformative for the field of aging research. The signaling pathways that control aging have been mapped out in considerable detail through research in various model organisms. The insulin and insulin-like growth factor pathway, sometimes called the IGF-1 pathway, has been shown to influence aging across a remarkable range of species, from worms and flies to mice and possibly humans. Reducing signaling through this pathway extends lifespan in various laboratory organisms, with the effects being substantial. The mechanism appears to involve effects on metabolism, stress resistance, and various cellular maintenance processes. Whether modulating this pathway in humans would have similar effects is being investigated, with various drugs and interventions that affect the pathway being studied. The mTOR pathway is another major signaling pathway that affects aging. The mTOR protein integrates signals about nutrient availability and growth conditions, regulating various cellular processes, including protein synthesis, autophagy, and cell growth. Reducing mTOR activity, either through genetic modifications or through drugs like rapamycin, extends lifespan in various species. The mechanism involves enhanced autophagy, improved stress resistance, and various other beneficial effects on cellular function. Rapamycin has become one of the most promising candidate interventions for human aging, with various clinical studies investigating its potential effects on human health and longevity. The NAD+ pathway, which involves a molecule called nicotinamide adenine dinucleotide that is essential for various cellular processes, has been the subject of substantial recent research. NAD+ levels decline with age in various tissues, with the decline contributing to dysfunction in multiple systems. Various interventions that boost NAD+ levels have shown promising effects in laboratory studies, with potential implications for slowing aging and treating various age-related conditions. The development of NAD+ precursors that can be taken as supplements, including nicotinamide riboside and nicotinamide mononucleotide, has produced significant commercial interest, though the actual benefits in humans remain to be definitively established. Stem cell biology provides another important perspective on aging. Stem cells are the cells that produce the various specialized cells of tissues, with their continued function being essential for tissue maintenance and repair. The populations of stem cells decline with age in most tissues, and the remaining stem cells often show reduced function compared to younger cells. The reduced capacity for tissue maintenance contributes to the various deteriorations that characterize aged tissues. Various approaches to addressing stem cell dysfunction in aging are being investigated, including approaches to enhance the function of existing stem cells and approaches to introduce new stem cells into aged tissues. The growing understanding of all these various mechanisms has produced a picture of aging that is genuinely impressive in its complexity. Aging is not a single process, but a complex network of interconnected mechanisms, each of which can, in principle, be studied and potentially modified. The interventions that target specific mechanisms have shown promising effects in laboratory studies, with various approaches being investigated for potential application to human aging. Whether the various approaches will eventually produce substantial extensions of healthy human lifespan, or whether the complexity of human aging will continue to resist effective intervention, remains to be seen. The research continues, with new discoveries continuing to emerge at a rapid pace. A single cell within your body, perhaps somewhere in one of your tissues, going about its various functions in this very moment. The cell is performing its specific role, whether it is contracting as a muscle cell, conducting electrical signals as a nerve cell, producing some specific protein as a liver cell, or any of the other specialized functions that the many cell types of your body perform. While it is performing its function, the cell is also engaged in continuous maintenance activities. Its protein quality control systems are working to identify and remove damaged proteins. Its DNA repair systems are working to fix various kinds of damage to its genetic material. Its autophagy machinery is clearing away damaged cellular components. Its mitochondria are producing energy while also generating some reactive oxygen species that contribute to ongoing damage. The telomeres at the ends of its chromosomes are slightly shorter than they were before the cell's last division. The epigenetic marks on its DNA are gradually shifting in the patterns that characterize aging. All of this is happening simultaneously in this single cell, and similar processes are happening in trillions of other cells throughout your body. The cumulative effect of all these processes, integrated across your entire body and continuing across the years and decades of your life, produces the patterns of aging that you experience. Tonight, like every night, the processes continue. The cells continue to maintain themselves, to accumulate damage, to enter senescence in some cases, to be replaced in others. The vast complex biology of your body continues its operations, with the aging process being woven into the very fabric of the maintenance and repair systems that keep you alive. The contemplation of this, on a quiet night like this one, is itself a kind of acknowledgement of the remarkable and mysterious biological reality that constitutes your existence. The various interventions that have been investigated for their potential to slow or reverse aging represent one of the most active areas of contemporary biomedical research, and the specific approaches that have shown the most promise deserve careful examination. The work in this area has produced both genuine breakthroughs and significant disappointments, with the picture remaining incomplete, but with sufficient progress to suggest that meaningful interventions in human aging may be possible within the lifetimes of people currently living. The careful walk through what has been tried, what has worked, and what remains uncertain provides one of the more interesting pictures available in modern biology. Caloric restriction stands as the most thoroughly studied intervention with demonstrated effects on aging across many species. The basic finding is that reducing food intake to approximately 70 or 80% of what an organism would naturally consume while maintaining adequate nutrition extends lifespan substantially in essentially every species in which it has been tested. The effect was first observed in rats in studies conducted in the 1930s, with researchers finding that animals fed restricted diets lived significantly longer than those allowed to eat freely. Subsequent studies have confirmed the effect in worms, flies, fish, mice, dogs, and various other species, with the lifespan extension typically ranging from 20 to 50%, depending on the species and the specific restriction protocol. The studies in non-human primates have produced particularly interesting results. Two major long-term studies of caloric restriction in rhesus monkeys, conducted at the University of Wisconsin and at the National Institute on Aging, have followed groups of monkeys for several decades under controlled feeding conditions. The Wisconsin study showed substantial reductions in age-related diseases and improvements in survival in the restricted monkeys compared to the controls. The NIA study showed less dramatic effects on survival, but did show significant improvements in various health measures. The differences between the two studies have been attributed to various factors, including differences in the control diets used and differences in the specific restriction protocols. The cumulative evidence from the primate study suggests that caloric restriction does produce real benefits for health and probably for lifespan in monkeys, though the magnitude of the effects is not as dramatic as the effects seen in shorter-lived species. Whether caloric restriction would extend human lifespan is a question that has been difficult to answer directly because of the practical challenges of conducting controlled studies of human longevity. Studies in humans have shown that caloric restriction produces various measurable benefits, including improvements in cardiovascular markers, reductions in inflammation, improvements in insulin sensitivity, and various other indicators of better health. The Calorie Study, a multi-year clinical trial of moderate caloric restriction in healthy adults, demonstrated that participants who maintained about a 15% reduction in caloric intake showed improvements in various health markers that have been associated with longer lifespan in other contexts. Whether these benefits will translate to actual lifespan extension in humans is not yet known, but the various indirect indicators suggest that some benefit is likely. The challenge with caloric restriction as a practical intervention for humans is that maintaining such restriction over decades is genuinely difficult. Most people find chronic caloric restriction unpleasant and unsustainable, with the constant hunger, reduced energy, and various other effects making long-term adherence challenging. Even motivated individuals who attempt sustained caloric restriction often find it difficult to maintain the level of restriction necessary to produce significant biological effects. The practical limitations of caloric restriction as a longevity intervention have motivated substantial research into compounds that might produce similar effects without requiring the difficult dietary changes. The discovery that various compounds can produce some of the same effects as caloric restriction without requiring the actual restriction has been one of the more exciting developments in recent aging research. These compounds, sometimes called caloric restriction mimetics, work by affecting some of the same biological pathways that caloric restriction activates. The most promising of these compounds have shown lifespan extending effects in laboratory studies, with various clinical investigations underway to determine whether similar effects occur in humans. Rapamycin has emerged as perhaps the most promising candidate compound for human longevity intervention. The drug was originally discovered in soil samples from Easter Island, with the substance being produced by bacteria that live in the soil. The original interest in rapamycin was as an immunosuppressant for organ transplant patients, where it has been used successfully for decades. The recognition that rapamycin also affects the mTOR pathway, which is centrally involved in aging, led to studies of its potential effects on lifespan. The results have been remarkable. Mice given rapamycin late in life, starting at an age equivalent to a human in their 60s, showed substantial lifespan extensions of around 10 to 20%. The effect was particularly notable because it occurred when the intervention began at relatively advanced ages, suggesting that rapamycin can produce benefits even when the aging process is already well underway. The mechanism by which rapamycin extends lifespan involves its effects on the mTOR signaling pathway. When mTOR activity is reduced, cells respond by enhancing autophagy, improving protein quality control, and activating various stress resistance pathways. The cumulative effect is improved cellular function and reduced accumulation of the various forms of damage that contribute to aging. Whether rapamycin will extend human lifespan in a similar way to its effects in mice is being investigated through various clinical studies, with the early results suggesting that the drug does produce various beneficial effects in humans, including improved immune function and various other markers of better health. The use of rapamycin in humans for longevity purposes is currently happening primarily through off-label prescribing, since the drug has not been formally approved for anti-aging applications. Various physicians have begun prescribing rapamycin in low doses for healthy patients who are interested in potential longevity benefits, with the doses being much lower than those used for transplant patients to minimize potential side effects. The actual effects of these protocols on human lifespan remain to be established, since the time needed to observe effects on longevity is necessarily long. The early data on various health markers in humans taking rapamycin have been encouraging enough to support continued investigation, though definitive conclusions await longer-term studies. Metformin represents another widely studied candidate for longevity intervention. The drug has been used for many decades to treat type 2 diabetes, with millions of people taking it daily without significant problems. The recognition that metformin might have anti-aging effects emerged from observations that diabetic patients taking metformin appeared to have lower rates of various age-related conditions, including some cancers, cardiovascular disease, and cognitive decline, compared to diabetic patients on other medications, and even compared to non-diabetic populations. The implications of these observations led to studies of metformin's potential mechanisms and to the proposal that the drug might have general anti-aging effects beyond its specific use in diabetes treatment. The mechanisms by which metformin might affect aging involve multiple pathways. The drug affects mitochondrial function, reducing the production of reactive oxygen species. It influences the AMPK pathway, which is involved in cellular energy sensing and various metabolic processes. It affects gut microbiota in various ways that may contribute to its overall effects. It influences various inflammatory pathways. The combination of these effects appears to produce beneficial outcomes that go beyond the drug's direct effects on glucose metabolism. The TAME study, a large clinical trial designed to evaluate metformin specifically for its anti-aging effects rather than its diabetes effects, is currently being organized to provide more definitive evidence about whether metformin extends healthy human lifespan. Senolytics represent a different category of intervention, focusing specifically on the removal of senescent cells from tissues. The development of senolytic drugs has progressed rapidly over the past decade, with various compounds being identified that selectively kill senescent cells while leaving healthy cells unaffected. The first generation of senolytics included compounds like dasatinib and quercetin, which when used in combination showed promising effects in mouse studies. Subsequent research has identified various other compounds with senolytic activity, and clinical trials are now investigating these drugs for potential applications in various age-related conditions. The early clinical trials of senolytics have produced encouraging results for specific applications. A study in patients with idiopathic pulmonary fibrosis, a fatal lung disease associated with cellular senescence, showed that intermittent dosing with dasatinib and quercetin produced improvements in physical function. Another study in patients with chronic kidney disease showed reductions in senescent cell burden and improvements in various functional measures. These early results have generated substantial interest in expanding the investigation of senolytics for a broader range of applications, with various clinical trials currently in progress. The genetic interventions for aging represent another major line of research, with various approaches being explored for potentially modifying the genetic factors that contribute to aging. The development of gene editing technologies, particularly CRISPR, has made it possible to consider interventions that would not have been feasible even a few years ago. Various researchers have been exploring potential genetic interventions that might address specific aspects of aging, including approaches to maintain telomere length, to remove or modify genes that contribute to age-related conditions, and to enhance the function of genes that support cellular health. The genetic interventions remain primarily at the research stage rather than at the clinical application stage, but the rapid development of the underlying technologies suggests that genetic approaches to aging may become practically relevant within coming decades. The parabiosis experiments represent a particularly fascinating line of research that has shed light on the role of blood factors in aging. Parabiosis involves surgically connecting the circulatory systems of two animals so that they share blood. When young and old mice are connected through parabiosis, the old mice show various improvements in tissue function, with effects that appear to result from factors in the young blood. The reverse situation also occurs, with the young mice showing some signs of accelerated aging from exposure to the old blood. The implications of these findings have been the subject of intense investigation, with researchers working to identify the specific factors in young blood that produce beneficial effects, and the specific factors in old blood that produce detrimental effects. The identification of specific blood factors that affect aging has been one of the major goals of parabiosis-inspired research. Various proteins have been identified that increase or decrease with age, and that appear to contribute to aging-related effects. The protein GDF11 was initially proposed as a key rejuvenating factor in young blood, though subsequent research has produced mixed results about its specific role. Other proteins, including various growth factors and hormones, have been investigated for similar potential effects. The identification of the specific molecular mediators of the parabiosis effects has been challenging, but the basic finding that blood composition affects aging-related processes has been confirmed across multiple studies. The practical implications of the blood factor research have led to various applications being investigated, including approaches to remove harmful factors from old blood and approaches to administer beneficial factors. Plasma exchange, in which a patient's plasma is removed and replaced with various replacement solutions, has been investigated for its potential effects on aging, though the actual benefits remain to be definitively established. Various commercial ventures have begun offering plasma transfusion services targeted at individuals interested in potential longevity benefits, though the scientific support for these specific applications is limited. The legitimate research continues to investigate the underlying biology, with the eventual translation to practical interventions depending on how clearly the specific mechanisms can be identified and targeted. Exercise and physical activity represent perhaps the most thoroughly established interventions for healthy aging, with the evidence for benefits being among the strongest in all of biomedical science. Regular physical activity is associated with reduced risk of essentially every major age-related disease, including cardiovascular disease, type 2 diabetes, various cancers, dementia, and many others. The mechanisms by which exercise produces these benefits involve multiple pathways, including effects on cardiovascular function, metabolic regulation, inflammation, neurogenesis, and various other processes. The specific exercise prescriptions that produce optimal benefits are still being refined, but the basic conclusion that regular physical activity contributes substantially to healthy aging is among the most robust findings in modern medicine. The various dietary approaches that have been investigated for potential aging effects represent another category of intervention. The Mediterranean diet, characterized by emphasis on plant foods, healthy fats, fish, and limited red meat consumption, has been associated with reduced rates of various age-related diseases in numerous observational studies. The specific dietary components and patterns that contribute to these effects are still being investigated, but the overall pattern of effects is consistent enough to support the dietary approach as a likely contributor to healthy aging. Other dietary approaches, including various forms of intermittent fasting, ketogenic diets, and plant-based diets, have all shown various health benefits, though the specific implications for aging are still being established. The development of artificial intelligence applications for aging research has accelerated the pace of discovery in recent years. Machine learning algorithms can analyze enormous data sets of biological information to identify patterns that would be difficult for human researchers to recognize. AI applications in drug discovery have already identified various potential aging-relevant compounds that are now being investigated through more traditional research methods. The integration of AI into aging research is likely to continue and to accelerate, with various applications including improved analysis of complex biological data, identification of new therapeutic targets, and personalization of interventions based on individual biological characteristics. A research laboratory somewhere in the world, perhaps in the middle of the night, with the lights on and various experiments running. Petri dishes contain cultures of cells being exposed to potential anti-aging compounds, with researchers monitoring how the cells respond. Mice in carefully controlled housing are receiving various treatments while their health and behavior are being monitored across months and years. Genetic sequencing machines are processing samples from human subjects in clinical trials, generating data about how various interventions affect biological markers of aging. Computer servers are running complex analyses of accumulated data, identifying patterns that might point toward effective interventions. The research continues in countless laboratories around the world, with each individual study contributing some increment to the cumulative understanding. The various researchers involved represent some of the most dedicated scientific workers in the world, with their careers devoted to understanding aging and to developing interventions that might extend healthy human lifespan. The work is patient and detailed, with many failures along the way and only occasional successes. But the cumulative progress has been substantial, and the pace of advancement has been accelerating. Whether the work will eventually produce interventions that substantially extend healthy human lifespan remains to be seen. But the investigation continues, with the various efforts gradually accumulating into a picture that may eventually transform how humans relate to the aging process. Tonight, like every night, the work proceeds. The cells grow in their dishes. The mice live their lives in their carefully controlled environments. The data accumulates on the servers. The researchers continue their patient investigations. The accumulated effort of all this work may eventually change what it means to grow old in ways that no individual researcher fully anticipates, but that the collective enterprise is gradually working toward. The relationship between aging and disease deserves careful attention, because most of what we recognize as the harmful aspects of aging are actually specific diseases that become more common with advancing age rather than direct consequences of the aging process itself. The distinction between aging and age-related disease is more subtle than it might initially seem, and understanding it has important implications for how we should think about interventions to improve health in older age. Cardiovascular disease is the leading cause of death in most modern societies, with rates increasing substantially with age. The conditions grouped under this category include coronary artery disease, in which the blood vessels supplying the heart become narrowed by accumulated deposits called atherosclerotic plaques, stroke, in which blood flow to part of the brain is interrupted, heart failure, in which the heart becomes unable to pump blood effectively, and various other conditions involving the cardiovascular system. The accumulation of arterial plaques is a process that begins relatively early in life, with detectable arterial changes being present in many people by middle age. But the clinical consequences typically do not become apparent until later in life, when the cumulative damage has progressed sufficiently to interfere with normal function. The biology of atherosclerosis involves multiple interconnected processes, including chronic inflammation, oxidative damage to lipids in the blood, dysfunction of the endothelial cells that line blood vessels, and the recruitment of various immune cells to the developing plaques. The plaques themselves contain not only the accumulated lipid deposits, but also various cells including smooth muscle cells, immune cells, and connective tissue components. The development of plaques can occur over decades, with periods of slow growth alternating with periods of more rapid progression, and the clinical consequences depending on whether plaques remain stable or undergo rupture that triggers acute events like heart attacks or strokes. The various interventions that reduce cardiovascular disease risk address different aspects of the underlying biology. Statins, the most widely prescribed cardiovascular medications, reduce the production of cholesterol in the liver and therefore reduce the lipid material available for plaque development. Antihypertensive medications reduce the mechanical stress on blood vessels by lowering blood pressure. Antiplatelet drugs reduce the tendency for blood clots to form on damaged arterial surfaces. Various lifestyle interventions, including exercise, dietary changes, and smoking cessation, address multiple aspects of the underlying biology and have substantial effects on cardiovascular risk. The cumulative effect of modern cardiovascular medicine has been substantial reductions in cardiovascular mortality in developed countries over the past several decades, contributing significantly to the overall increases in life expectancy that have occurred. Cancer represents another major category of age-related disease, with risk increasing dramatically with advancing age. The fundamental basis for cancer is the accumulation of genetic mutations that disrupt the normal control mechanisms regulating cell division. Most cells contain various protective mechanisms that prevent the development of cancer, including DNA repair systems that fix mutations, tumor suppressor genes that prevent inappropriate cell division, and immune surveillance that identifies and eliminates abnormal cells. The progressive failure of these protective mechanisms with age contributes to the increasing cancer risk in older populations. The relationship between aging and cancer is particularly interesting because of the various ways that aging biology and cancer biology intersect. The shortening of telomeres that contributes to cellular senescence can also limit the ability of cells to undergo the indefinite proliferation that characterizes cancer. Cancer cells frequently activate telomerase to overcome this limitation, allowing them to continue dividing beyond the normal limits. The cellular senescence that we discussed earlier serves multiple functions, with one of its important roles being the prevention of cancer through the elimination of cells that have accumulated dangerous mutations. The complex relationships between aging mechanisms and cancer prevention mean that interventions targeting aging biology need to be evaluated carefully for their potential effects on cancer risk. The treatment of cancer has advanced dramatically over recent decades, with various approaches, including improved surgical techniques, more targeted radiation therapy, conventional chemotherapy, and the newer categories of targeted therapies and immunotherapies, producing substantial improvements in outcomes for many cancer types. The development of immunotherapies in particular, which work by enabling the immune system to recognize and attack cancer cells, has transformed the treatment of various previously difficult cancers. The continued progress in cancer treatment, combined with improved methods for early detection of cancer when it is more treatable, has substantially improved cancer survival rates over the past several decades. Neurodegenerative diseases represent a particularly challenging category of age-related conditions. Alzheimer's disease and related dementias affect tens of millions of people worldwide, with the numbers expected to increase substantially as populations age. The fundamental biology of Alzheimer's disease involves the accumulation of two types of protein aggregates in brain tissue: the amyloid plaques composed of beta-amyloid protein and the neurofibrillary tangles composed of tau protein. The development of these aggregates appears to begin decades before clinical symptoms appear, with the gradual accumulation eventually causing sufficient brain dysfunction to produce the cognitive and behavioral changes that characterize the disease. The treatment of Alzheimer's disease has been particularly challenging, with multiple attempts to develop effective therapies producing limited success. The recent approval of antibody-based treatments that target amyloid in the brain represents a potential turning point, though the actual benefits of these treatments in terms of clinical outcomes have been modest, and they carry significant risks, including various brain abnormalities. The complexity of Alzheimer's disease biology, with multiple interacting mechanisms contributing to the overall pathology, means that effective treatment may require addressing multiple aspects simultaneously rather than focusing on any single target. Parkinson's disease involves the loss of neurons in specific brain regions, particularly those producing the neurotransmitter dopamine, with the resulting deficits producing the characteristic motor symptoms including tremor, slowness of movement, and impaired balance. The underlying biology involves the accumulation of aggregates of the protein alpha-synuclein, with the protein pathology gradually spreading through various brain regions over time. The treatment of Parkinson's disease has been more successful than the treatment of Alzheimer's, with dopamine replacement therapy and various other approaches providing significant symptom relief for many patients. However, the underlying disease process continues to progress, and there are currently no treatments that effectively halt or reverse the neurodegeneration. Various other neurodegenerative conditions, including frontotemporal dementia, Lewy body dementia, amyotrophic lateral sclerosis, and various more rare conditions, share common features of progressive neuronal loss driven by the accumulation of misfolded proteins. The shared underlying biology has led to substantial interest in approaches that might address protein aggregation across multiple conditions, though the specific manifestations of different diseases reflect the different proteins involved and the different brain regions affected. The continued investigation of neurodegenerative biology is among the most active areas of medical research, with the urgency reinforced by the substantial human costs of these conditions and the increasing prevalence as populations age. The musculoskeletal conditions that affect older populations represent another major category of age-related health problems. Osteoporosis, the progressive loss of bone density that occurs particularly in postmenopausal women, but also in older men, contributes to substantial fracture risk in older populations. The biology of osteoporosis involves an imbalance between the cells that build bone and the cells that resorb bone, with the net effect being gradual loss of bone tissue over time. Various treatments including bisphosphonates, hormone therapies, and newer biological agents can substantially reduce the rate of bone loss and reduce fracture risk in affected individuals. Sarcopenia, the progressive loss of muscle mass and strength that occurs with aging, has been increasingly recognized as an important contributor to disability and mortality in older populations. The biology of sarcopenia involves multiple factors, including reduced protein synthesis, impaired regenerative capacity of muscle tissue, chronic inflammation, and various hormonal changes. The most effective interventions for sarcopenia are resistance exercise combined with adequate protein intake, with these approaches having been shown to substantially reduce muscle loss and even reverse some of the changes in older adults who adopt them. The development of pharmaceutical interventions for sarcopenia has been less successful, though various approaches are being investigated. Osteoarthritis, the most common form of arthritis, involves the progressive degradation of the cartilage that cushions joints, leading to pain and reduced function. The condition affects most older adults to some degree, with the severity varying considerably across individuals. The biology of osteoarthritis involves complex interactions between mechanical stress, inflammation, and various biological factors that affect cartilage maintenance and repair. The current treatments focus primarily on symptom management through pain medications, physical therapy, and in severe cases, joint replacement surgery. The development of disease-modifying treatments that would address the underlying cartilage degradation has been challenging, though various approaches including cell-based therapies and biological agents are being investigated. Type 2 diabetes represents another major category of age-related condition, with rates increasing substantially in recent decades, partly because of population aging and partly because of various other factors, including dietary changes and reduced physical activity. The condition involves impaired regulation of blood glucose, with the fundamental problem being either insufficient insulin production by the pancreas or resistance to insulin action in peripheral tissues. The complications of diabetes include accelerated cardiovascular disease, kidney disease, nerve damage, eye disease, and various other problems that contribute to substantial reductions in quality of life and life expectancy. The treatment of type 2 diabetes has improved substantially in recent decades, with various medications and lifestyle approaches that can effectively manage the condition for most patients. The newer categories of medications, including GLP-1 agonists, have shown particularly promising effects on diabetes outcomes and have additional benefits for weight management and cardiovascular health. The vision and hearing problems that increase with age affect quality of life substantially, even when they do not directly threaten survival. Age-related macular degeneration, cataracts, and various other eye conditions can substantially impair vision in older adults. Age-related hearing loss similarly affects many older adults and contributes to social isolation, cognitive decline, and various other problems. The treatments for various age-related vision problems have improved substantially, with cataract surgery being one of the most successful medical interventions ever developed, and various treatments now available for macular degeneration that can substantially slow disease progression. The treatments for hearing loss, including modern hearing aids and cochlear implants, have similarly improved, though many older adults with hearing loss do not access these treatments for various reasons. The relationship between mental health and aging deserves attention as well, as depression and anxiety affect substantial proportions of older adults, with the rates being influenced by factors including health problems, social isolation, loss of loved ones, and various other circumstances. The treatment of mental health problems in older adults is essentially similar to treatment in younger populations, with various medications and therapeutic approaches being effective. However, mental health problems in older adults are sometimes underdiagnosed and undertreated, with various stigmas and practical barriers contributing to inadequate care. The recognition of mental health as a crucial component of healthy aging has been growing, with various efforts to improve identification and treatment of mental health problems in older populations. The concept of health span has become increasingly important in discussions about aging interventions. Health span refers to the period of life spent in good health, free of significant disability or disease, in contrast to lifespan, which refers to total length of life. The focus on extending health span, rather than just lifespan, reflects the recognition that simply living longer is not necessarily desirable if the additional years are characterized by significant disability or suffering. The most desirable outcome from aging interventions would be extension of the healthy years of life, with the period of disability and disease being compressed into a shorter portion of the total lifespan, ideally with relatively rapid decline at the end of life rather than extended periods of progressive disability. The compression of morbidity hypothesis, originally proposed by James Fries in the 1980s, suggested that this kind of outcome might be achievable through various interventions. The basic idea is that if the age of onset of various chronic diseases can be delayed more than the age of death can be extended, the period of chronic illness would be compressed into a shorter portion of life. Various data have been examined to assess whether this kind of compression has been occurring in recent decades, with mixed results. Some evidence suggests that disability rates have been declining in some populations, supporting the compression hypothesis. Other evidence suggests that the gains in life expectancy have not been accompanied by proportional reductions in disability years, contradicting the simple compression model. The actual pattern probably varies across populations and time periods, with the specific outcomes depending on multiple factors, including healthcare access, lifestyle factors, and the specific medical conditions involved. A medical research facility somewhere in the world, with various clinical trials in progress, investigating different interventions for age-related diseases. Patients with various conditions are participating in studies that may eventually lead to better treatments or even cures for the conditions that currently limit healthy aging. Researchers analyze data from these studies, trying to understand which interventions work, for which patients, and through which mechanisms. The work proceeds incrementally, with each successful study adding some increment to the cumulative understanding, and each failed study eliminating one possibility from consideration. The patients who participate in these studies contribute their bodies and their time to the collective enterprise of advancing medical knowledge, with most of them receiving uncertain personal benefit, but contributing to the eventual development of better treatments for future patients. The slow accumulation of medical knowledge has produced dramatic improvements in the treatment of many age-related conditions over recent decades, with continued progress expected to produce further improvements in coming years. The ongoing investigation continues in countless research settings, with the cumulative effect gradually changing what it means to grow old in the 21st century. Tonight, like every night, the research proceeds. The trials continue. The data accumulates. The understanding gradually deepens. The future of how humans experience aging is being shaped in this very moment by the patient work of researchers and clinicians around the world. The diversity of human aging deserves attention because the variation across individuals is far greater than the average patterns suggest. Two people of the same chronological age can be in dramatically different states of health, function, and biological aging. The factors that contribute to this diversity include genetic differences, lifestyle factors, environmental exposures, social circumstances, and various other influences that combine to produce the specific aging trajectory that each individual experiences. Understanding this diversity is essential for understanding aging as it actually occurs in humans rather than as an abstract average process. The genetic contribution to longevity has been investigated extensively, with various studies attempting to identify the specific genetic variants that affect how long people live and how well they age. The conclusion from this research is that genetics plays a substantial role in determining longevity, but the role is more complex than the role of genetics in many other traits. Studies of twins have estimated that genetics accounts for approximately 20 to 30% of the variation in human lifespan, with the remainder being attributable to environmental and lifestyle factors. This heritability is lower than for many traits, suggesting that environmental and behavioral factors play substantial roles in determining how long people live. Studies of exceptionally long-lived individuals, including centenarians who live to 100 or beyond, and supercentenarians who live to 110 or beyond, have provided particularly interesting insights into the genetics of human longevity. These exceptionally long-lived individuals appear to differ genetically from the general population in various ways, with several specific genetic variants being more common among centenarians than among shorter-lived people. The APOE gene, which is involved in lipid metabolism and is also a major risk factor for Alzheimer's disease, has variants that affect longevity, with certain forms being more common among centenarians and other forms being associated with shorter lifespan. The FOXO3 gene, which is involved in various cellular stress response pathways, has variants associated with exceptional longevity. Various other genetic factors have been identified that appear to contribute to long life, though the specific contributions of individual variants are often modest. The concept of biological age as distinct from chronological age has become increasingly important in modern aging research. Two people who are 70 years old by the calendar can be in biologically very different states, with one having the physiological function of a typical 55-year-old and another having the physiological function of an 80-year-old. The factors that produce this divergence between chronological and biological age include the various genetic, environmental, and lifestyle factors that affect aging biology. The ability to measure biological age through various biomarkers, including epigenetic clocks, telomere length, inflammatory markers, and various other indicators, has provided new tools for understanding individual variation in aging. The epigenetic clocks that we discussed earlier have proven particularly useful for assessing biological age. The patterns of DNA methylation that change predictably with age allow estimation of biological age with remarkable accuracy. People whose biological age, as measured by these clocks, is greater than their chronological age tend to have higher mortality risk and higher rates of various age-related diseases. People whose biological age is lower than their chronological age tend to have better health outcomes. The ability to measure biological aging objectively has been transformative for aging research, providing a way to assess whether interventions actually affect the underlying aging process rather than just affecting specific health indicators. The lifestyle factors that affect aging deserve careful attention because they represent the aspects of aging that individuals can most directly influence. The cumulative evidence about which lifestyle factors most substantially affect aging outcomes has become reasonably clear, with several patterns consistently emerging across many studies. Regular physical activity is associated with substantial benefits for essentially every aspect of aging, with the effects being among the strongest and most reproducible in all of medical science. Adequate sleep contributes to numerous aspects of healthy aging, with chronic sleep deprivation being associated with various negative outcomes. Stress management and social connection both contribute to better aging outcomes, with chronic stress and social isolation being associated with accelerated aging. Avoiding tobacco and limiting alcohol consumption contributes substantially to better outcomes. The cumulative effect of these various lifestyle factors can be very substantial, with people who maintain favorable patterns across multiple domains often showing dramatically better aging outcomes than those who do not. The blue zones, regions of the world where exceptional longevity is more common than in surrounding regions, have been studied extensively for clues about lifestyle factors that support healthy aging. These regions include Okinawa in Japan, Sardinia in Italy, the Nicoya Peninsula in Costa Rica, Icaria in Greece, and the Seventh-day Adventist community in Loma Linda, California. The populations in these regions share various features, including diets that emphasize plant foods, regular natural physical activity integrated into daily life, strong social connections, sense of purpose, and various other factors. The specific contributions of each factor are difficult to disentangle, but the cumulative effects appear to produce the exceptional longevity patterns that characterize these regions. The dietary patterns associated with the blue zones share various features that are worth noting. The diets typically emphasize plant foods including vegetables, fruits, legumes, whole grains, and nuts. Meat consumption tends to be moderate to low, with various plant proteins providing substantial portions of dietary protein. Fish consumption is often a feature in coastal blue zones. Olive oil and other healthy fats are typically included. Refined sugars and processed foods are typically minimal. The patterns are not identical across the various blue zones, but they share enough features to suggest that the general approach of emphasizing whole plant foods with moderate amounts of other items supports healthy aging. The physical activity patterns in the blue zones are also interesting. The activity tends to be integrated into daily life rather than being formal exercise. People walk extensively, garden, perform manual household tasks, and engage in various other activities that involve consistent low to moderate intensity physical effort. The patterns differ substantially from the formal exercise regimens that are common in modern developed societies, with the natural integration of activity into daily life producing the cumulative effects rather than dedicated exercise sessions. The implications for thinking about physical activity prescriptions are interesting, suggesting that consistent moderate activity may be at least as important as intense formal exercise. The social and psychological factors that characterize the blue zones deserve special attention. Strong social connections, including close family relationships, integration into community life, and various forms of mutual support, are consistent features. The sense of purpose, called ikigai in Okinawa, and similar concepts in other regions, refers to having reasons to get up in the morning and engage with the world. The participation in religious or spiritual communities, regardless of specific tradition, has been associated with longer life in various studies. The relatively low stress levels and the cultural patterns that support emotional well-being contribute to the overall picture. The cumulative effect of these psychosocial factors appears to be substantial, suggesting that human flourishing involves much more than just physical health. The role of social connection in aging has been investigated extensively in recent decades, with the cumulative evidence supporting its importance becoming increasingly clear. Social isolation has been shown to have effects on mortality comparable in magnitude to smoking, obesity, and other major risk factors. The mechanisms by which social connection affects aging involve multiple pathways, including effects on stress hormones, inflammation, mental health, and various behavioral factors. The implications for aging policy and individual decisions are substantial, with the maintenance of social connections being recognized as an essential component of healthy aging. The differences in aging patterns across human populations have been extensively documented. Some populations show consistently better aging outcomes than others, with various factors contributing to the differences. Genetic factors play some role, but environmental and lifestyle factors appear to be more important in most cases. The differences in life expectancy across countries have changed substantially over time, with developing countries generally catching up to developed countries as healthcare access and public health measures have improved. Within countries, substantial differences in life expectancy exist across different demographic groups, with the differences reflecting factors including socioeconomic status, access to healthcare, environmental exposures, and various other influences. The historical changes in human longevity over recent centuries deserve attention because they illustrate how dramatically the human experience of aging has shifted. For most of human history, life expectancy at birth was substantially shorter than it is today, primarily because of high infant and child mortality. Once individuals survived childhood, their life expectancy was somewhat closer to modern figures, though still typically shorter. The dramatic increases in life expectancy that have occurred over the past century or so have been driven primarily by reductions in infant mortality, improvements in nutrition, advances in medical care, and various other public health improvements. The continued increases in life expectancy in developed countries during recent decades have been driven more by reductions in mortality at older ages than by reductions in childhood mortality, since childhood mortality is already very low in these populations. The current life expectancy in most developed countries is approximately 80 years, with some countries reaching the mid-80s. The historical trajectory has been steadily upward, with each generation typically living somewhat longer than the previous generation. Whether this trend will continue, accelerate, or reverse is genuinely uncertain. Various factors, including potential improvements in medical care, could continue to increase life expectancy, while various other factors, including increasing obesity rates, climate change effects, and various social problems, could potentially work against further increases. The actual trajectory will depend on how these various factors interact over coming decades. The differences in aging patterns by sex deserve specific attention. Women generally live longer than men in essentially all human populations, with the average difference being about 4 to 7 years in most developed countries. The factors that contribute to this difference include both biological factors and behavioral factors. Women tend to have various biological characteristics that may contribute to longer life, including different hormonal patterns, different patterns of cardiovascular disease risk, and various other differences. Women also tend to engage in fewer behaviors associated with shorter life, including being less likely to smoke heavily, less likely to die in accidents, and less likely to die from violence. The specific contributions of biological versus behavioral factors are difficult to disentangle precisely, but both appear to contribute to the observed differences. The variation in aging within individuals across different organ systems and tissues is also worth understanding. The same individual can have one organ system that is aging relatively rapidly, while another system is aging relatively slowly. This intra-individual variation in aging means that the overall picture of an individual's aging is complex, with different systems showing different patterns of decline. The clinical implications of this variation are significant, with the prediction of various health outcomes requiring assessment of multiple systems rather than relying on any single indicator. The development of more comprehensive approaches to assessing individual aging trajectories, incorporating multiple biological systems, is an active area of research. The psychological experience of aging varies enormously across individuals and cultures. Some people experience the aging process with relative equanimity, accepting the changes that occur and finding meaning and satisfaction in later life. Others experience aging with substantial distress, struggling with the various losses and changes that occur. The factors that affect how aging is experienced psychologically include cultural attitudes about aging, individual personality characteristics, social circumstances, health status, and various other influences. The cultural attitudes about aging have substantial effects on how individuals experience the process, with cultures that respect and value older people generally producing better psychological outcomes than cultures that devalue or marginalize older people. The cultural variation in how aging is experienced and valued is itself a fascinating topic. Some cultures, particularly various traditional cultures and some Asian cultures, have historically valued older people highly and integrated them centrally into family and community life. Other cultures, particularly many modern Western cultures, have tended to marginalize older people and emphasize youth as the most valued life stage. The effects of these cultural differences on the actual experience of aging are substantial, with people in cultures that value older people generally experiencing the aging process with more dignity and satisfaction than people in cultures that do not. The implications for how societies should think about aging are significant, suggesting that cultural and social attitudes are themselves important factors in determining aging outcomes. A small village in one of the Blue Zone regions, perhaps in Sardinia or Okinawa, in the early hours of the morning. The older residents of the village are beginning their daily routines, with the patterns being essentially similar to what their grandparents did at similar ages decades ago. Some are tending small gardens, harvesting vegetables for the day's meals. Others are walking through the village to visit friends or to perform various community functions. The integration of these older individuals into the daily life of the village is complete, with their contributions being valued and their presence being normal rather than exceptional. The diets they will eat today consist primarily of vegetables from their gardens, with modest amounts of olive oil, fish, beans, and grains. The social connections they will maintain today include multiple interactions with family members, neighbors, and friends. The sense of purpose they experience involves their continuing contributions to their families and communities, with their accumulated wisdom and experience being valued by those around them. The pattern of life that supports their exceptional longevity is not a special intervention, but simply the way that life has always been lived in their community. Tonight, when the day ends, they will sleep comfortably in their homes, surrounded by the social and physical environment that has supported their long lives. The cumulative effect of all these specific features of their existence has been to allow many of them to reach exceptional ages while maintaining good health and meaningful connection to their world. The model that they collectively represent is not directly transferable to modern industrialized societies, but it provides important evidence about what kinds of conditions support healthy human aging. The contemplation of this model, on a quiet night like this one, is itself a kind of reflection on what aging could look like under different conditions than those that characterize most modern lives. The brain and cognitive aspects of aging deserve substantial attention because they affect not just how long people live, but how they experience their existence as they grow older. The brain undergoes various changes with age, with some changes being relatively benign and others contributing to significant cognitive decline. Understanding what happens to the aging brain and what factors influence the trajectory of cognitive aging has been one of the most active areas of neuroscience research in recent decades. The structural changes that occur in the aging brain are now reasonably well documented. The brain gradually loses volume with age, with the loss occurring at different rates in different regions. The prefrontal cortex, which is involved in executive function, planning, and various complex cognitive operations, tends to show particularly substantial volume loss in older age. The hippocampus, which is essential for forming new memories, also shows significant age-related changes. Other brain regions show smaller but still measurable changes. The volume losses reflect various processes, including some loss of neurons, reduction in the density of connections between neurons, changes in the white matter that connects different brain regions, and various other structural alterations. The functional changes in the aging brain occur in parallel with the structural changes. Various cognitive functions show characteristic patterns of change with age. Processing speed, the rate at which the brain can perform various cognitive operations, declines fairly consistently with age starting from young adulthood. Working memory, the ability to hold and manipulate information temporarily, also shows age-related declines. Episodic memory, the ability to remember specific events and experiences, similarly declines with age. Executive function, including planning, decision-making, and inhibition of inappropriate responses, shows age-related changes that contribute to various practical difficulties in older age. However, the picture of cognitive aging is not uniformly negative. Various cognitive abilities are relatively preserved or even continue to improve with age. Vocabulary and general knowledge typically continue to expand throughout most of adult life, with older adults often having larger vocabularies and broader factual knowledge than younger adults. Various forms of practical wisdom, the ability to apply accumulated knowledge to real-world problems, tend to develop with age and experience. Emotional regulation often improves with age, with older adults frequently showing better ability to manage their emotional responses to various situations. The patterns of cognitive aging are therefore complex, with some abilities declining while others are maintained or improved. The factors that influence cognitive aging have been investigated extensively, with various findings now reasonably well established. Physical exercise has substantial benefits for cognitive aging, with regular aerobic exercise being associated with reduced rates of cognitive decline and reduced risk of dementia. The mechanisms involve effects on brain blood flow, neurogenesis in the hippocampus, reduction of inflammation, and various other pathways. Cognitive engagement throughout life appears to contribute to better cognitive aging, with people who maintain intellectually stimulating activities generally showing better cognitive function in older age. Social engagement similarly contributes to cognitive health, with social isolation being associated with accelerated cognitive decline. Adequate sleep is essential for cognitive health, with various studies showing that chronic sleep problems contribute to cognitive decline and dementia risk. The relationship between sleep and cognitive aging deserves specific attention because of recent research that has clarified important mechanisms. The brain has a specialized waste clearance system called the glymphatic system, which removes various metabolic byproducts including proteins associated with neurodegenerative diseases. The glymphatic system operates most actively during sleep, with the clearance being substantially enhanced during deep sleep stages. The implications are significant. Chronic sleep deprivation or poor sleep quality may impair the brain's ability to clear away proteins like beta amyloid that contribute to Alzheimer's disease. The connection between sleep quality and dementia risk that has been observed in various epidemiological studies may operate through this glymphatic clearance mechanism. The maintenance of good sleep throughout life may therefore be one of the most important factors for long-term cognitive health. The concept of cognitive reserve has become important in understanding why some people maintain cognitive function better than others as they age. Cognitive reserve refers to the brain's capacity to maintain function despite various forms of damage or aging. People with greater cognitive reserve can sustain more brain pathology before showing clinical symptoms of cognitive decline. The factors that build cognitive reserve include education, intellectually demanding work, complex social engagement, and various forms of mental activity throughout life. The implications are significant because they suggest that the cumulative effects of intellectual engagement throughout life can affect cognitive outcomes decades later. The development of dementia, and specifically Alzheimer's disease, represents one of the most feared aspects of aging for many people. The biology of Alzheimer's disease involves multiple processes that develop over decades before clinical symptoms appear. The accumulation of beta-amyloid plaques in brain tissue is one of the earliest detectable changes, with the plaques beginning to develop in some individuals as early as middle age. The development of tau neurofibrillary tangles tends to occur somewhat later and to spread through specific brain regions in characteristic patterns. The combination of these protein pathologies eventually produces sufficient brain dysfunction to cause clinical symptoms, but the underlying biology has typically been developing for many years before the symptoms become apparent. The treatment options for Alzheimer's disease have improved somewhat in recent years, though the available treatments remain limited. The cholinesterase inhibitors and memantine that have been used for decades provide modest symptomatic benefits for some patients, but do not affect the underlying disease process. The newer antibody-based treatments that target beta-amyloid in the brain have shown some ability to slow disease progression in clinical trials, though the magnitude of the clinical benefits has been modest and the treatments carry significant risks. The development of effective disease-modifying treatments for Alzheimer's remains one of the most important goals in modern medical research, with various approaches being investigated. The prevention of cognitive decline has become an important focus of research and public health efforts. The evidence supports several specific strategies that appear to reduce the risk of cognitive decline and dementia. Regular physical activity is among the most strongly supported strategies. A diet emphasizing plant foods, fish, and healthy fats appears to provide benefits. Maintaining cognitive engagement through various forms of intellectual activity contributes to better outcomes. Social engagement throughout life is important. Adequate sleep, stress management, treatment of various health conditions, including hypertension and diabetes, and avoiding head injuries, all contribute to better cognitive aging. The cumulative effect of maintaining favorable patterns across these various domains can be substantial, with various studies suggesting that adherence to comprehensive healthy lifestyle patterns may reduce dementia risk by 30 to 50% or more. The phenomenon, sometimes called successful aging or healthy aging, has been studied extensively, with researchers trying to identify what allows some people to age with relatively preserved function, while others experience substantial decline. The cumulative picture is that successful aging involves multiple interacting factors operating across the entire lifespan. Genetic factors play some role, but environmental, lifestyle, and social factors play substantial roles. The patterns established in early and middle adulthood appear to have lasting effects on later life outcomes, with the cumulative effects of various positive and negative influences building up over decades. The role of purpose and meaning in healthy aging deserves specific attention. Various studies have shown that having a sense of purpose in life is associated with better health outcomes, including lower mortality risk, reduced rates of various diseases, and better cognitive function in older age. The mechanisms by which purpose affects health are not fully understood. but they probably involve effects on stress responses, motivation to engage in healthy behaviors, social connections, and various other pathways. The implications for thinking about aging are significant. The maintenance of meaningful engagement with life, with purposes that extend beyond immediate self-concerns, appears to be one of the important contributors to healthy aging. The relationship between aging and creativity has been investigated, with various findings being relevant to how we should think about cognitive aging. While some forms of creativity, particularly those requiring rapid processing and novel insight, may peak in younger adulthood, other forms of creativity continue to develop throughout life. The work of older artists, writers, scientists, and other creative individuals often demonstrates that significant creative contributions can be made throughout adult life. The patterns vary across different domains, with some fields tending toward earlier creative peaks and others toward later peaks. The general point is that aging does not eliminate creative capacity, though it may transform the kinds of creativity that individuals can effectively pursue. The accumulation of expertise that occurs over decades of professional and personal life represents one of the genuine benefits of aging. Older individuals in various fields typically have deeper knowledge of their domains, better practical judgment, and more refined intuitions about complex situations than younger individuals. The expertise that develops over a lifetime cannot be acquired in any shorter time, and the contributions that experienced individuals can make often differ qualitatively from those that younger individuals can make. The value of accumulated expertise has been increasingly recognized in various professional contexts, with the contributions of older workers and mentors being valued in ways that go beyond simple measures of productivity. The mental health aspects of aging deserve careful consideration. Depression and anxiety affect substantial proportions of older adults, with the rates being influenced by various factors, including health problems, social losses, and life transitions. The treatment of mental health problems in older adults is essentially similar to treatment in younger populations, with various medications and therapeutic approaches being effective. However, mental health problems in older adults are sometimes underdiagnosed and undertreated, with various stigmas and practical barriers contributing to inadequate care. The recognition that mental health is an essential component of aging well has been growing, with various efforts to improve identification and treatment of mental health problems in older populations. The psychological adaptations that some older adults make to the various challenges of aging are worth understanding. Research has shown that many older adults achieve what psychologists call gerotranscendence, a shift in perspective that involves greater acceptance of mortality, less concern with material success and superficial social relationships, and greater appreciation for nature, spirituality, and meaningful connections. This shift, when it occurs, can produce substantial well-being in older age, despite various physical and social losses. The psychological capacity for this kind of adaptation appears to be one of the genuine strengths that human development can produce. The relationship between aging and time perception is itself fascinating. Many older adults report that time seems to pass more quickly as they age, with years feeling shorter than they did during youth and middle age. Various theories have been proposed to explain this phenomenon, including the proportional theory, which suggests that each unit of time represents a smaller fraction of total life experience as one ages, and various theories involving changes in attention and memory consolidation. The subjective experience of time, however explained, is part of how aging is actually experienced, and the contemplation of this aspect of aging is itself a kind of reflection on the nature of temporal existence. The end-of-life issues that become more relevant in older age deserve at least brief mention. Most people, when they consider their own aging, eventually have to confront questions about how they want to live in their final years, how they want to die, what kinds of medical interventions they want or do not want, and various other questions about the conclusion of life. The medical and social systems for supporting people through end of life have developed substantially in recent decades, with hospice care, palliative medicine, and various other approaches providing options for compassionate care that were not previously available. The conversations about end of life that families and individuals can have in advance of medical crises help to ensure that the wishes of dying individuals are respected and that the experience of dying is as good as it can be under the inherent difficulties of the situation. Hold this in mind as you continue to drift. An older person, perhaps in their 80s, sitting quietly in their home in the evening hours. The day has been ordinary, with various activities completed, conversations held, simple pleasures experienced. The person has experienced losses over the years, friends and family members who have died, capabilities that have declined, situations that have changed irretrievably. But the person has also experienced the accumulation of wisdom, the deepening of certain relationships, the development of perspectives that were not possible during younger years. The mind is somewhat different from what it was decades ago, with certain capacities reduced, but with other capacities preserved or even enhanced. The body operates within the constraints of its current condition, with various adaptations having been made to accommodate the changes that have occurred. The person sitting in this room is the same person who was once a young child, who was once a young adult, full of ambitions and uncertainties, who was once a middle-aged person, engaged in the various projects of mature life. The continuity of identity across all these stages is one of the more remarkable features of human existence, with each person carrying within themselves the accumulated history of all the previous versions of themselves that have led to this current moment. Tonight, like every night, older people around the world are sitting in their homes, contemplating their lives, experiencing the specific quality of consciousness that comes with having lived for many decades. The contemplation of this universal experience on a quiet night like this one is itself a kind of acknowledgement of the remarkable continuity that connects each of us across all the stages of our possible lives. The frontier research on radical life extension represents some of the most ambitious and controversial work being conducted in the science of aging. The basic question being investigated is whether the various mechanisms of aging that have been identified can be modified sufficiently to produce dramatic extensions of human lifespan beyond what currently seems possible. The work spans multiple approaches and includes both established research institutions and various less conventional ventures, with the cumulative effect being a substantial expansion of what is being investigated and what may eventually become possible. The fundamental question that motivates this research is whether human lifespan has a fixed upper limit, or whether the current maximum reflects only the limits of current biology that could potentially be modified. The longest documented human lifespan is currently held by Jeanne Calment, a French woman who lived to 122 years, with various other supercentenarians having reached ages over 115. Whether the maximum human lifespan is fixed at something like 120 to 125 years, or whether interventions might eventually allow substantially longer lives, is genuinely uncertain. Some researchers believe that the apparent ceiling reflects fundamental biological limits that will be difficult or impossible to exceed. Others believe that the ceiling reflects only the current state of biology and that substantial extensions may be possible with appropriate interventions. The cellular reprogramming approaches that have been developed in recent years represent one of the most exciting areas of frontier aging research. The basic discovery underlying this approach was made by Shinya Yamanaka, who showed in 2006 that mature cells could be reprogrammed back to a pluripotent state similar to embryonic stem cells through the introduction of just 4 specific genes, now called the Yamanaka factors. The implications of this discovery for aging research emerged when researchers recognized that the reprogramming process appeared to reverse various indicators of cellular age, including epigenetic patterns associated with aging. The question that has been investigated is whether partial reprogramming, using the Yamanaka factors briefly enough to reverse aging-related changes without fully resetting cells to a pluripotent state, might provide a method for rejuvenating aged tissues. Studies in mice have produced remarkable results in this area. Researchers have shown that brief exposure of aged mouse tissues to the Yamanaka factors can reverse various biomarkers of aging, restore function to aged organs, and extend lifespan in certain experimental conditions. The work has been impressive enough to attract substantial commercial investment, with various biotech companies working to develop partial reprogramming approaches for human applications. The challenges of translating the mouse work to human applications are substantial, including risks of inappropriate cellular changes that could lead to cancer and various other potential problems. But the proof of principle that aged tissues can be rejuvenated through reprogramming approaches has been firmly established, and the development of safe and effective human applications is a major focus of ongoing research. The Altos Labs initiative, launched in 2022 with substantial investment from various sources, including Jeff Bezos and other major investors, represents one of the largest and most ambitious efforts in this area. The company has recruited many of the leading researchers in cellular reprogramming and aging biology, with the goal of developing therapies that could substantially extend healthy human lifespan through cellular rejuvenation approaches. The scale of the investment and the caliber of the scientific team have made Altos one of the most watched ventures in aging research. Whether the company will eventually produce successful therapies remains to be seen, but the level of commitment to the approach suggests that substantial progress may be possible. The work of David Sinclair at Harvard Medical School has been particularly influential in this area. Sinclair has been investigating the role of NAD and the sirtuin proteins in aging for many years, and his work has contributed to the understanding of how cellular metabolism affects aging. More recently, his laboratory has been investigating cellular reprogramming approaches, with various findings suggesting that the epigenetic patterns associated with aging can be reversed through appropriate interventions. Sinclair has also been a prominent public communicator about aging research, with his book Lifespan presenting both the scientific evidence and his personal views about the prospects for human life extension. His views have been controversial, with some other researchers being skeptical of his more optimistic claims, but his work has helped to bring substantial attention to the possibilities of intervening in aging biology. The investigation of senolytics has continued to advance, with various new compounds being developed and clinical trials expanding to investigate effects on a broader range of conditions. The basic principle of removing senescent cells from tissues has been validated in numerous studies, with the potential applications extending beyond simple lifespan extension to include treatment of various age-related diseases. The clinical translation of senolytic therapies is still in relatively early stages, with much work remaining to identify the optimal compounds, dosing regimens, and patient populations. But the proof of principle that selective elimination of senescent cells can produce health benefits has been firmly established, and continued development of senolytic therapies is one of the more promising areas of clinical translation. The development of various gene therapies for aging-related conditions represents another major frontier. The technologies for delivering genetic material to specific cells and tissues have advanced substantially, making it increasingly feasible to consider gene therapies that might address various aspects of aging. Approaches being investigated include therapies to maintain telomere length, to enhance the function of various cellular maintenance systems, to replace genes whose function has been compromised by mutations, and various other applications. The development of safe and effective gene therapies for aging is challenging because of the complexity of the underlying biology and the various potential side effects. But the rapid advancement of the underlying technologies suggests that genetic approaches to aging may become practically relevant within coming decades. The work on combining multiple interventions in aging represents an important recognition that single interventions may not be sufficient to produce dramatic effects on aging. The various mechanisms of aging are interconnected, with damage in one system contributing to dysfunction in others. Effective intervention may therefore require targeting multiple mechanisms simultaneously, rather than focusing on any single target. Various research programs are investigating combinations of interventions, including combinations of caloric restriction mimetics with senolytics, combinations of various supplements with lifestyle interventions, and various other multi-pronged approaches. The complexity of designing and evaluating multi-intervention protocols is substantial, but the cumulative evidence suggests that comprehensive approaches may produce greater benefits than any single intervention alone. The cryopreservation research that has been pursued by various organizations represents a particularly speculative approach to the problem of mortality. Cryopreservation involves cooling the body to very low temperatures shortly after legal death, with the intention of preserving the biological structure in a state from which the person might potentially be revived in the future when more advanced medical technologies become available. The various cryonics organizations have been preserving people for several decades, with the current population of cryopreserved individuals, including hundreds of people who have made the choice to be preserved after death. Whether any of these individuals can ever be successfully revived is genuinely uncertain. The challenges include not just the development of revival technologies, but also the question of whether the preservation process maintains the biological information necessary for the preserved individuals to retain their identities and memories. The scientific support for the eventual feasibility of revival is debated, with some researchers viewing it as essentially impossible and others viewing it as plausible given sufficient future technological advancement. The transhumanist perspectives that motivate some aging research deserve mention. Transhumanism is a philosophical movement that advocates for using technology to enhance human capabilities and overcome various biological limitations, including aging. The proponents of transhumanism argue that there is nothing inherently sacred about human biology in its current form, and that ethical considerations should lead us to develop technologies that could substantially improve the human condition, including dramatic extensions of healthy lifespan. The movement has produced various detailed proposals about how human enhancement might proceed and what the implications of various technological developments might be. The transhumanist perspectives are controversial, with various religious and philosophical traditions raising concerns about whether such enhancements would be desirable or wise. But the perspectives have influenced thinking about aging research and have motivated some of the more ambitious efforts to develop life extension technologies. The whole brain emulation research represents perhaps the most radical approach to the problem of mortality. The basic concept involves the eventual development of technology that could scan the structure and information content of a human brain in sufficient detail to create a functional replica of that brain in some other medium, possibly a computer system. The replicated mind would, in principle, retain the memories, personality, and other psychological characteristics of the original person, providing a kind of continuation of identity beyond the limits of biological existence. The technical challenges involved in such an approach are enormous, with the development of adequate scanning technologies, sufficient computational resources, and adequate understanding of brain function all being far beyond current capabilities. Whether such an approach will ever become feasible is genuinely uncertain, but the various researchers and organizations working on aspects of the problem suggest that some people view it as a serious long-term goal rather than as pure science fiction. The philosophical questions that arise from these various frontier approaches deserve substantial attention. If interventions become available that could substantially extend healthy human lifespan, various ethical and social questions become urgent. The question of who would have access to such interventions, with concerns about inequality being substantial. The question of how extended lifespans would affect population dynamics, with potential implications for resource use, environmental sustainability, and various other concerns. The question of how extended lifespans would affect social institutions, with concerns about generational turnover, career patterns, and various other social structures. The question of whether dramatically extended lifespans would actually be desirable from the perspective of the individuals living them, with concerns about whether longer lives would necessarily be better lives. These various questions do not have simple answers, and they deserve careful consideration as the technologies that might enable life extension continue to develop. The economic implications of extending human lifespan are substantial. If interventions become available that could keep people healthy and productive into ages substantially beyond the current normal lifespan, the implications for retirement systems, healthcare systems, labor markets, and various other economic structures would be profound. The systems that currently exist were designed around assumptions about typical human lifespan that may become increasingly outdated if effective life extension interventions become available. The economic transitions involved would be substantial, with various adjustments being required across multiple domains. The specific shape of these transitions would depend on how rapidly life extension technologies become available, how widely they become accessible, and how individuals and societies adapt to the new possibilities. The cultural implications of substantially extending human lifespan would also be significant. Various cultural patterns are organized around assumptions about typical lifespan, including family structures, career trajectories, generational relationships, and various other patterns. If lifespans become substantially longer, these cultural patterns would likely need to evolve in ways that are difficult to fully anticipate. The implications for how individuals understand the meaning and structure of their own lives would also be significant. The cultural and existential adjustments to dramatic life extension would probably be as substantial as the medical achievements that enable the extensions, with the cumulative effect being a transformation of what it means to be human. The current consensus among researchers in the field of aging is that substantial extensions of healthy human lifespan are likely achievable within coming decades, though the specific magnitudes and timeframes are uncertain. The interventions currently in development could potentially add years or even decades to healthy human lifespan if they prove effective in human applications. The more dramatic possibilities of doubling or more dramatically extending human lifespan would require more substantial advances, possibly including approaches like cellular reprogramming that are still in early development. Whether such dramatic extensions will eventually prove possible remains an open question. The investigation continues, with the various lines of research gradually accumulating into a picture that may eventually transform what aging means for the human species. The pace of advancement in aging research has been accelerating in recent years, with various developments contributing to optimism about the prospects for meaningful interventions. The improved understanding of aging biology, the development of better tools for measuring aging, including the epigenetic clocks, the substantial commercial investment in the field, the recruitment of talented researchers to work on aging problems, the development of various promising interventions, and the cumulative knowledge being gathered from clinical trials all contribute to a situation where the prospects for advancement are better than they have ever been. Whether the progress will continue at the current pace or will encounter obstacles that slow advancement remains to be seen. But the current trajectory suggests that significant developments in aging research are likely in the coming years and decades. A research facility somewhere in the world, perhaps one of the major aging research centers in the United States or Europe, with various experiments running and researchers analyzing data. The work being conducted in this facility, multiplied across many similar facilities around the world, represents the cumulative human effort to understand and potentially modify the biological process that has limited human lifespan throughout the entire history of our species. The researchers involved are working on problems that, if eventually solved, could fundamentally transform the human experience. Some are studying cellular reprogramming, trying to understand how the epigenetic patterns of aged cells can be safely reset to younger states. Others are developing senolytic compounds, testing various molecules for their ability to selectively eliminate senescent cells while leaving healthy cells unaffected. Others are conducting clinical trials of various potential anti-aging interventions, gathering the human data needed to determine whether the laboratory findings translate to actual benefits for people. Others are analyzing massive data sets of biological information, looking for patterns that might point toward new therapeutic targets. The cumulative effect of all this work, distributed across many research groups around the world, is substantial. Each individual project contributes some increment to the overall enterprise. The progress, when viewed over the span of just the past few decades, has been remarkable. Whether the progress will eventually produce interventions that substantially extend healthy human lifespan remains to be seen, but the investigation continues with energy and dedication. The scientists and clinicians involved are devoting their careers to the possibility that the limits of human lifespan can be substantially modified, and their cumulative work is gradually transforming our understanding of what may be possible. Tonight, like every night, the research proceeds. The cells in their dishes are being exposed to various potential interventions. The data on the servers is being analyzed for patterns. The patients in clinical trials are participating in studies that may contribute to future advances. The patient accumulation of knowledge and capability that characterizes scientific progress continues to operate in the specific domain of aging research, as in all other areas of inquiry. The eventual outcomes remain uncertain, but the process of investigation continues in the patient way that has produced essentially all of the medical advances that have transformed human existence over the past century. The philosophical and ethical questions raised by the prospect of extending human lifespan deserve careful examination, because these questions are not merely abstract concerns, but have practical implications for how we should think about aging research and the broader project of medical advancement. The questions touch on some of the deepest issues in human existence, including the meaning of life, the nature of personal identity, the ethics of distributing scarce resources, and the relationship between individual desires and broader social considerations. The fundamental question of whether dramatically extending human lifespan would be a good thing is more contested than it might initially appear. The intuitive response for most people is that longer life is obviously desirable, with the desire to continue living being one of the most basic human motivations. But the question becomes more complex when examined carefully. Would living for 200 or 300 years actually be a richer and more satisfying experience than living for 80 or 100 years? Various philosophers and thinkers have argued that there are good reasons to question whether radically extended lifespans would actually be desirable, even setting aside questions about whether they would be achievable. The argument that very long lives might not be desirable has been articulated by various thinkers, with the philosopher Bernard Williams providing one of the more influential analyses. Williams argued in an essay published in 1973 that human life eventually exhausts its meaningful possibilities, with the projects, relationships, and engagements that give life value being limited in their capacity to sustain indefinite interest. After a sufficiently long time, according to this argument, individuals would face a choice between maintaining the basic character that defines them as the persons they are, in which case, life would eventually become tediously repetitive, and undergoing such substantial personal change that the resulting being would no longer be recognizable as the same person who began the extended lifespan. Williams concluded that there are reasonable grounds to doubt that radical life extension would actually be desirable, even if it were achievable. The counter-arguments to Williams have been articulated by various other thinkers. The basic counter-argument is that human life offers essentially unlimited possibilities for engagement, learning, relationships, and meaningful activity. Even very long lives could continue to be enriched by new experiences, new relationships, new fields of study, and new forms of engagement with the world. The argument that life eventually exhausts its meaningful possibilities, according to this view, reflects a failure of imagination rather than a genuine limitation of what life can offer. People who live long lives well, according to this view, continue to find their existence meaningful and engaging, with the duration of life not being a fundamental constraint on its quality. The actual evidence from extremely long-lived individuals, while limited because of how few people reach extreme ages, generally suggests that the people who reach very old ages while maintaining reasonable health and function continue to find value in their existence. The various centenarians and supercentenarians who have been studied generally report continued satisfaction with life, continued meaningful relationships, and continued engagement with the world. The evidence from these individuals does not definitively settle the philosophical question, but it does suggest that the pessimistic predictions about life eventually becoming meaningless are not consistently borne out in practice. The question of personal identity over very long lifespans deserves careful examination. The philosophical question of what makes a person at one time the same person as a person at a later time has been debated for centuries, with various theories being proposed. The most common modern view emphasizes continuity of psychological characteristics, including memories, personality traits, beliefs, and values. By this measure, an individual whose psychology gradually evolves over a long lifetime would remain the same person despite the changes, as long as the changes occur gradually rather than abruptly. However, over very long timescales, the cumulative changes in any individual would be substantial, with the person at age 200 potentially having very different memories, beliefs, and characteristics than the person at age 30. Whether such an individual should be considered the same person across the full time span is a question that admits of different answers depending on which theory of personal identity is adopted. The implications of dramatic life extension for societies have been considered in various analyses, with both potential benefits and potential problems being identified. The benefits include the continued contributions of experienced individuals to various fields, with the accumulated wisdom and expertise of long-lived people potentially providing substantial value. The continued personal relationships across longer time spans could allow for deeper and richer connections than are possible with shorter lifespans. The continuity of personal projects and engagements could allow for more substantial individual contributions to various fields than current lifespans permit. The potential problems include various concerns about how dramatically extended lifespans would affect societies. Population dynamics would change substantially if individuals lived much longer, with implications for population growth, resource use, environmental sustainability, and various other factors. Career patterns would need to change, with the assumption that people work for 40 or 50 years and then retire, becoming inappropriate for substantially longer lives. The generational turnover that has historically allowed societies to change and adapt could slow if older generations continue to occupy positions of influence for longer periods. The relationships between generations could become strained if the assumption that older people eventually make way for younger people no longer applies. These various concerns do not have simple solutions, and addressing them would require substantial changes in social structures and arrangements. The question of access to potential life extension technologies raises significant equity concerns. If interventions become available that could substantially extend healthy lifespan, the distribution of these interventions becomes a major issue. If access is limited to wealthy individuals, while others continue to age and die at current rates, the resulting inequality would be unprecedented in its magnitude. Differences in lifespan based on wealth already exist and are considered problematic, but the differences would become much more substantial if some people live to 150, while others continue to die at 70 or 80. The various proposals for addressing these equity concerns include treating life extension technologies as essential health services that should be universally accessible, regulating the development and distribution of such technologies, and various other approaches. The actual policies that will develop around these technologies will depend on political and social processes that are difficult to predict in advance. The environmental implications of dramatic life extension deserve consideration. If human populations grow substantially because of longer lifespans, the increased pressure on environmental systems could be significant. The resources required to support larger populations, including food, water, energy, and various other inputs, would need to be either expanded or distributed more efficiently. The environmental impacts of larger human populations could be problematic if not adequately addressed. Whether the various environmental and resource challenges associated with population growth would be manageable depends on technological, social, and political developments that are uncertain. The question of whether substantially extending human lifespan is environmentally sustainable is therefore one of the genuine concerns that need to be considered in evaluating life extension research. The religious and spiritual perspectives on life extension deserve attention because various traditions have developed elaborate frameworks for thinking about death, mortality, and the meaning of human existence. The traditional religious perspectives generally view death as a natural part of human existence, with various traditions emphasizing the importance of accepting mortality and finding meaning within the limited time span of human life. The development of technologies that might substantially extend lifespan raises questions about how these traditional perspectives should be modified or maintained. Some religious thinkers have argued that life extension research should be supported because it represents a legitimate use of human capabilities to reduce suffering and extend the opportunity for meaningful existence. Others have argued that the pursuit of dramatic life extension represents a problematic effort to escape the natural limits of human existence, with potentially negative consequences for individual character and social arrangements. The various traditions have differed in their specific views about whether longer life is inherently desirable. Some religious traditions, particularly some forms of Buddhism, have emphasized that extended life is not necessarily desirable, with the suffering associated with continued existence in the world being one of the reasons that liberation from the cycle of rebirth has been considered a worthy goal. Other traditions have generally treated longer life as positive, with various promises of extended or eternal life appearing in the eschatologies of multiple traditions. The cumulative picture from religious thought about life extension is complex, with various perspectives offering different views about whether the pursuit of substantially longer life is wise or unwise. The question of how individuals should personally relate to the prospect of life extension is itself worth considering. The current generation of people interested in aging research includes both those who view life extension as a personal goal that they hope to benefit from, and those who are content with the typical human lifespan and view aging research as primarily beneficial for future generations. The personal decisions that individuals make about their own engagement with potential life extension technologies will depend on various factors, including their values, their assessments of the risks and benefits, their economic resources, and their willingness to be early adopters of unproven interventions. The decisions are individual rather than collective, with each person needing to determine for themselves how they want to relate to the various possibilities. The death and dying that current humans experience deserves attention, even in the context of life extension research. Even if substantial life extension becomes possible, death will eventually occur for all individuals, with the various processes of dying continuing to be features of human existence. The improvements in how dying is managed, including the development of hospice care, palliative medicine, and various other approaches, have been substantial in recent decades, but remain incomplete. The quality of death and dying for many people could be substantially improved through better integration of palliative approaches, better communication between patients and healthcare providers, and various other improvements. The attention to how people die well, even within the context of efforts to extend life, is an important aspect of how we should think about aging and mortality. The psychological adaptations that humans make to their finite lifespans are interesting to consider in the context of potential life extension. The recognition of mortality has been one of the central features of human consciousness, with various religious, philosophical, and cultural traditions developing in part as responses to the awareness of death. The contemplation of mortality has been seen by many traditions as essential to living well, with the recognition that life is limited, contributing to the appreciation of its value. Whether the elimination or dramatic extension of mortality would affect these psychological and cultural patterns in beneficial or detrimental ways is uncertain. Some thinkers have argued that the recognition of mortality is essential to human meaning, and that life extension would diminish the significance of existence. Others have argued that the elimination or reduction of mortality would free humans to engage with life more fully without the constant shadow of approaching death. The relationship between aging research and broader medical research deserves consideration. The investigation of aging biology has implications for the treatment of various age-related diseases, including Alzheimer's, cancer, cardiovascular disease, and many others. The benefits of aging research extend beyond simple lifespan extension to include potential improvements in the treatment of essentially every condition that becomes more common with age. The case for aging research can therefore be made independently of any specific claims about radical life extension, with the various potential benefits for human health and welfare being substantial even if dramatic lifespan extension proves to be unachievable. The framing of aging research in terms of health span extension and disease prevention may be more appropriate than the framing in terms of life extension, with the broader benefits being more widely beneficial and less subject to the various concerns about radical extension. The reasonable response to the prospects of aging research probably involves taking the work seriously without overstating either the immediate prospects or the eventual implications. The research is producing genuine advances that are likely to provide real benefits for human health in coming years and decades. The development of effective treatments for various age-related conditions is likely to continue, with the cumulative effect being improvements in healthy lifespan, even if dramatic increases in maximum lifespan are not achieved. The pursuit of more ambitious goals, including substantial extensions of healthy lifespan and possibly maximum lifespan, continues, with various promising lines of research being pursued. The eventual outcomes remain uncertain, but the work is genuinely consequential and deserves continued support and attention. The questions about how individuals should personally engage with aging research are essentially personal decisions that each person needs to make based on their own values and circumstances. The various lifestyle interventions that are reasonably well supported by evidence, including regular exercise, healthy diet, adequate sleep, stress management, and social engagement, are essentially universally beneficial and worth pursuing, regardless of one's views about more aggressive life extension efforts. The various medical interventions that are available for managing age-related conditions, including treatment of hypertension, diabetes, high cholesterol, and various other conditions, are similarly beneficial and worth pursuing for most people. The more speculative interventions, including various supplements, off-label use of drugs like rapamycin or metformin, and various other approaches require more individual judgment, with the evidence varying in strength and the risks varying across approaches. A philosopher's study somewhere in the world, perhaps in a university or in a quiet home office, with books on shelves containing the accumulated thought of many traditions about the questions that aging and mortality raise for human existence. The philosopher, perhaps in the late hours of the evening, is contemplating the questions that the development of life extension technologies poses for traditional ways of thinking about human existence. The questions are genuinely difficult, with various reasonable positions being defensible. The philosopher knows that the development of effective life extension technologies will require not just scientific advances, but also careful thought about the implications and the ways that human existence might need to be reconceived if substantial extensions of lifespan become possible. The work being done in laboratories around the world is changing what is biologically possible, but the broader questions about whether the changes are desirable, how they should be distributed, and how individual and social life should adapt to them are questions that require the kind of careful reflection that philosophy and ethics provide. The philosopher will continue working on these questions, contributing to the broader conversation about how humans should respond to the prospects that the scientific work is opening up. The conversation will continue across many years and decades, with the eventual conclusions emerging from the cumulative thinking of many people in many traditions. Tonight, like every night, the careful contemplation of these questions continues in various places around the world, with the cumulative effect gradually shaping how humans understand their own existence and their relationship with the biological process of aging that has defined human life throughout our species' history. The closing part of this long exploration arrives now, and the cumulative weight of everything that has been examined tonight points toward a picture of aging that is genuinely more complex and more interesting than the casual encounter with this universal human experience would suggest. The picture has implications worth carrying with you as the night settles into its final hours. The walk through aging across these many parts has been patient and detailed. The exploration began with the basic question of what aging actually is, finding that it is not simply the passage of time, but a specific biological process with specific mechanisms operating at the molecular and cellular levels. The evolutionary explanations for why aging exists at all were considered, with the recognition that aging reflects the priorities that natural selection has favored, rather than being an inevitable feature of life itself. The remarkable diversity of aging patterns across species was examined, with some organisms showing essentially no signs of aging and others aging extremely rapidly. The specific cellular and molecular mechanisms that contribute to human aging were explored in detail, including cellular senescence, telomere shortening, protein aggregation, mitochondrial dysfunction, and various other processes. The various interventions that have been investigated for their potential effects on aging were examined, with some showing genuine promise and others remaining speculative. The relationship between aging and age-related diseases was considered, with the recognition that much of what we recognize as the harmful aspects of aging consists of specific diseases that become more common with age rather than direct consequences of aging itself. The diversity of human aging trajectories was examined, with the substantial variation across individuals reflecting genetic, environmental, and lifestyle factors. The brain and cognitive aspects of aging were explored. The frontier research on radical life extension was considered. The philosophical and ethical questions raised by the prospect of substantially extending human lifespan were addressed. Each of these topics, taken individually, is interesting. Taken together, they form a picture worth articulating carefully now. The picture is that aging is not a single inevitable process that humans must simply accept, but a complex biological phenomenon that is increasingly being understood and that may eventually be substantially modified through deliberate intervention. The picture is also that the cumulative effect of various lifestyle factors that individuals can influence is substantial, with the maintenance of healthy patterns across multiple domains producing better aging outcomes than would otherwise occur. The picture is further that the broader questions raised by the prospect of life extension are genuinely difficult, with various reasonable positions being defensible and no simple answers being available. This recognition has implications worth sitting with carefully. The first implication is that we live in a genuinely interesting moment in the history of human engagement with aging. The scientific understanding of aging has advanced dramatically in recent decades, with the identification of specific mechanisms that produce aging and the development of various interventions that might modify these mechanisms. The pace of advancement has been accelerating, with continued progress likely in the coming years. We are participants in a period of human history when the relationship between humans and the aging process may be undergoing substantial transformation, with the eventual outcomes being uncertain but potentially significant. The second implication concerns the relationship between individual decisions and aging outcomes. The various lifestyle factors that have been shown to substantially affect aging outcomes are largely within individual control. The maintenance of regular physical activity, adequate sleep, healthy nutrition, social engagement, stress management, and various other patterns produces substantial benefits for healthy aging. The cumulative effect of maintaining favorable patterns across multiple domains can be very substantial, with various estimates suggesting that healthy lifestyle patterns can effectively add many years of healthy life expectancy. The implications for personal decisions are significant. While many aspects of aging are beyond individual control, the patterns of daily life that individuals adopt have substantial effects on how the aging process unfolds for them. The third implication concerns the relationship between aging research and the broader human enterprise. The investigation of aging is one of the genuinely consequential scientific projects being pursued in our time, with the potential to produce dramatic improvements in human welfare. The continued support for this research, both through public funding and through private investment, is likely to produce substantial benefits for current and future generations. The research is not just about extending lifespan, but about addressing the various diseases and disabilities that limit healthy aging, with the potential benefits being widely distributed across the human population. The fourth implication, worth leaving you with as the exploration comes to its close, concerns how to think about your own aging in the context of what has been examined. You are engaged right now in the process of aging that has been the subject of this exploration. Your cells continue to undergo the various changes that have been described. Your tissues continue to accumulate the various forms of damage that contribute to aging. Your biology continues its slow trajectory toward the eventual ending that all biological organisms experience. The contemplation of this reality, on a quiet night like this one, is itself a kind of acknowledgement of your participation in the universal human experience of growing older. The reasonable response to this reality involves multiple elements. The acceptance of aging as a fundamental aspect of biological existence, without unnecessary resistance or denial. The active engagement with the lifestyle factors that can substantially improve aging outcomes, including the maintenance of healthy patterns across the various domains that have been shown to matter. The reasonable attention to medical care that can address various age-related conditions when they develop. The cultivation of psychological and spiritual capacities that support well-being throughout life, including in older age. The maintenance of meaningful engagement with the world through relationships, activities, and purposes that provide ongoing reasons for continued investment in life. The cumulative effect of these various elements is a relationship with aging that is neither resigned acceptance nor desperate resistance, but rather a mature engagement with one of the fundamental features of human existence. There is something almost spiritual about this when sitting with it carefully, although the language need not be pushed too far in any specific direction. The traditional wisdom traditions have generally recognized that engagement with mortality and aging is one of the central tasks of human life, with the cultivation of appropriate relationships with these universal features being essential to human flourishing. The modern scientific understanding of aging does not diminish the relevance of this traditional wisdom, but rather complements it, providing additional tools and perspectives for engaging with the fundamental realities that humans have always faced. The combination of scientific understanding and traditional wisdom can produce a richer engagement with aging than either alone would support. One final thought before the exploration concludes. The story of aging in the 21st century is not yet finished. The research continues, with new discoveries continuing to emerge at a rapid pace. The interventions that may eventually become available continue to be developed and tested. The cultural and social responses to aging continue to evolve. The personal decisions that individuals make about how to engage with their own aging continue to shape their experience. The future of aging, both as a biological process and as a human experience, remains genuinely open. The decisions and developments of the coming decades will shape what aging means for future generations in ways that are not yet determined. You and I are participants in this ongoing story, contributing in our various ways to how it unfolds. The contributions are small individually, but cumulatively significant, with the collective effect being one of the major themes of human existence in this period of history. If you have found this exploration valuable, if the patient walk through the complex story of aging has given you something to carry with you as you finally drift off, please consider letting me know in the comments below. Tell me what struck you most, what questions you would like to explore further, what other deep features of human existence you would like to spend a long, quiet night thinking about. Subscribe to Sleepy Tinfoil Hat if the channel is the kind of thing you want more of, because the channel exists for the audience that wants this slow, careful kind of thinking, and the audience tells the algorithm we are here through the small acts of comment and subscription. The community of careful nighttime thinkers is scattered across many time zones and many lives, and the way we find each other and keep this work going is through these small signals. Sleep well. Let yourself go gently. The biological processes that have been the subject of this exploration continue to operate in your body as you drift toward unconsciousness. Your cells continue their various maintenance activities. Your various biological systems continue their functions. The aging process continues in its slow, steady way, regardless of whether you attend to it. The processes will continue through the night and into the morning. They will continue through tomorrow and the days that follow. They will continue throughout the remainder of your life, in whatever specific way your life unfolds. The aging that you are engaged in is the same aging that every human who has ever lived has been engaged in. The continuity is real, even though each individual life has its own specific trajectory. You are part of the vast human experience of growing older, contributing your specific instance to the universal pattern that has characterized human existence throughout our species' history. Tonight, like every night, you are joining the long human practice of moving forward through time with all the biological and experiential changes that this involves. The practice is dignified by all the previous humans who have engaged in it before you, and it will continue to be the practice of future humans long after you are gone. The contemplation of this, on a quiet night like this one, is one of the small ways that humans can be more conscious participants in their own biological existence, in the patient way that suits a long, quiet evening drawing toward its close.